process this data. We take the entire protein and do one loop replacement at a time, and then it will all go into help, because since there are 4 serotypes, for each of the serotypes we can then have a tetravalent vaccine. One of them might stand for serotype 1. This second part would stand for the serotype 2, 3, 4, and so on. And every time we are making sure that the AB loop and FL loop is not as it is in the envelope protein, just so that the ADE won't be happening. And in doing that, we can recognize there are computational pipelines here. What are the pipelines? We should, first of all, collect available viral sequence genome data from different patient samples, not from the 1940s to 1960s, all the while until 2025. We can do that, but then we give a lot of variations, so the consensus becomes very noisy. So instead, we focused on the last 5 years alone, viral genome data, and then we created some consensus sequences, which is like an average. A residue is observed and retained a lot in one place, so we don't want to disturb it. Whereas in some regions where other changes already going on, we will make changes there. Okay, so once we identify a sequence, we perform what is called modeling, which is nothing but 3-dimensional modeling. And nowadays you would have heard of this software called AlphaFold, for which the Nobel Prize was given. How many of you know about AlphaFold a little bit already? You have heard of AlphaFold? Please raise your hands. Okay, so some of you know. Okay, AlphaFold uses artificial intelligence, and given the primary structure of a protein, it will provide a 3-dimensional solution. So we can use modeling and followed by energy minimization. That is to make sure that the protein model is at the best energetic state. And then we perform what are called as structure validation, which is to make sure there are no strains in the backbone polypeptide, and then ensuring that the antigenicity is very high, because by mutating we can make it worse. So we don't want that to happen. And then it's also checked using independent other softwares, and we want to make sure that the epitope binds to the antibody of interest. And this can be achieved by means of what is called as protein-protein docking, because the one protein is the envelope E protein of the dengue, the other protein is the antibody we are talking about, which comes from the host. So these two are proteins that have to dock to each other without any covalent bonds. So that is also achieved, and that's shown here. This is the overall computational pipeline, and we start with the sequence, take a consensus from there, and then we build a multiple sequence alignment to make sure which regions are not touched, so we don't want to change that. And then only the variable parts are modified. Okay, and that's first part of the pipeline, where we create the consensus. And then the second part of the pipeline is, after you get a 3-dimensional model, perform energy minimization and do what is called a structure validation. And I'm going to show you one example here. This is the sequence on top. This is the primary structure of the envelope protein, except that the AG strands are kept the same. That is the one that we saw in green and violet, which will interact with which antibody? Important antibody I kept mentioning. I mentioned like 4 times. What is the antibody? 4E11. Yes, because after that you will have a quiz. You please listen very, very carefully. You really want to get some prize, right? Okay. The 4E11 antibody you do not want to change, so therefore the AG strands are all kept the same, but the AB loop and FL loop has changed, and those are shown in sort of this red and orange color. You can see they have gone through the change as consulting the Discotope software. And what is Discotope? Is discovering alternate epitope regions. So we discussed that. Okay. Once we get this sort of alternate sequence, it goes through a 3-dimensional modeling using AlphaFold or our own lab where I did my postdoc. It is called as Modeler. You can use either of the softwares. You get a 3-dimensional model, do minimization, and then perform the structure validation. What is this? It provides a 2-dimensional map where the different backbone torsion angles phi and psi from the model are all read off and mapped onto the allowed regions of the Ramachandran map. This is a very good contribution. It's a very nice contribution from India, And if you don't know, it's okay, but I would urge you to go and read about this Ramachandran map. So the more they are within these allowed regions, yellow and red color, if you see points within that, it's a good model. And you can see that there are very few which are wandering around in the disallowed regions. So suggesting that it is a sound model, it's not a trained model. And then we also have independent methods, and this is called PROSA. If someone is interested, you can go read up, so I will skip it in the interest of time. So these are the structure validations. And then the antigenicity, because a correct antibody should bind, and the wrong antibodies, what is wrong antibodies? Those which provides this cross-reactive ADEs should not bind. That we discussed. Remember the analogy on the exams I was telling you. You should focus and read and not get into other distractions. So also here, we want the right antibodies to bind and the wrong antibodies not to bind. So that gets tested in this part of the pipeline. And then subsequently, independently, the immunogenicity is also checked. Okay, we'll quickly go to this part. This AG region and the green region should be conserved, and it should bind to 4E11. Whereas this AB loop and FL loop should not bind. So that is the objective. Let's see how well these modeled and that sequence did. We found that 100% of the residues that is supposed to bind to the 4E11 antibody is indeed binding. Just like how it has shown in the wild type, 44 out of 44 residues are in fact binding to this mutated sequence. Why should we worry? Because you might think, after all, we are doing small changes in the AB loop and FL loop. Why it should affect? Of course, it will bind. That might be an inherent assumption, but it is not true, because you have not only done modeling, you also done energy minimization, where you're essentially allowing the whole model to relax. Plus, my postdoc who has done all of this work, Dr. Palomi Ray, she has also done what is called as a molecular dynamics, wherein every atom is given a force and it's allowed to move within the constraints it will move. So that means the protein is constantly wiggling. When we say protein, we mean the viral E protein. Therefore, you can see whatever happens in the AB loop and FL loop, although it is far from the antibody binding region, the epitope region, there can be allosteric effects by which alterations can happen. And we found this for some of the other mutants as well. But here you can see 44%, 100% of the residues are indeed maintained. So the next question is, how good are we in masking the other regions? After all, we have modified these loops. So for that, independently, other antibodies which are known to bind to the AB and FL loop, such as these, are first of all dock. These are protein-protein docking programs by which we want to make sure that they don't bind that well. And you can see that compared to the wild type now, only 19% of the residues, 5 out of 26 residues are binding to the AB loop. And in this case, the FL loop. And the next slide will show us how it is in the AB loop, where also there are changes that are happening. Here, only 1 out of 15 residues that are known to bind in the wild type is now binding to the antibody, which is something else. It's not 4E11, it's some other antibody which is responsible for this ADE and the cross non-neutralizing antibodies. So we can see that from the computational point of view, we have achieved what we wanted. We wanted the classical regions to bind. Yes, they are binding, 100% of the residues are participating, but those residues, the loop regions, AB and FL loop regions where it's not supposed to bind, the interactions have reduced. That is a good sign. Okay, so we do have a wet lab biologist, Dr. Sweta Raghavan, who's testing this out. She comes from my collaborator's lab, but now she has formed her own company, so wherein she's going to take these alternate mutant E protein of the dengue virus, and she's going to challenge it to the human sera, and then see whether the antibodies are indeed being produced. We want the 4E11 antibodies to be higher in number, but not the other two antibodies, so that truth will come once we start to put all this into the wet lab practice and validation. But there is another tool that we use. We make sure that the epitope regions are all preserved by means of using another independent tool, which is called discontinuous B cell epitope, because the total domain itself was discontinuous. And even the lateral ridge, we saw that some parts were green, some parts were violet, and they come from different beta strands. So these are definitely called as discontinuous epitopes. These are also called as structural epitopes, so they are much harder to deal with. Luckily, we did not have to change that region, only the AV loop and FL loop which were short. Okay, so this is to show that the antigenicity profile is still there. So with this, I will stop and summarize what we have tried to do. What I have presented now is only for the DEN-V1 protein, but we are simultaneously also looking at the other 3 serotypes. We have built mutant models, and we have recognized optimal and the best mutant solutions for each of the serotypes. And right now, as we speak, just yesterday I had got a trainee who has come from a different college, and she's going to now do a very systematic survey and give all possible amino acid changes, as it were, to the AB and FL loop regions, and she'll build a matrix and identify which combinations of the ABFL loops are optimal. Because we seem to be lucky to have landed with this, but how does it do for the different 4 serotypes also could be a question. And why this will all be relevant is because then we can form the tetravalent vaccine, whereas the DENG 1, 2, 3, 4 will be there. All of them will be the mutant type. And all of them, AB and FL loops will have been altered. So hopefully the AD problem should not arise. And this is a very long way to go. But these are the things that we have tried to do so far. And these are the people behind. Dr. Paulomi Ray, she was trained. Okay, anyway, this was the last slide. It's okay. Dr. Paulomi Ray, she was trained in the US, and in fact, she has practiced, and she's an expert in developmental biology in different labs. And she joined our lab to learn bioinformatics, and from day one, she has picked it up and done all of these work. And in fact, she's working on another project and also done equally well. And plus, she's in a 3rd group project. Yeah, that's what happens in our lab. If once they come, they seldom stay with only one project. They usually work on 3 or 4 projects, and at least 2 of them will be teamwork. For example, I did not talk about Ayurvedic database that we have built, where the entire lab was involved, and she also played a role. And then the other person whom you can see in this picture is Dr. Sweta Raghavan, whom I talked about. This is the wet lab person who had already done some vaccine design for dengue, but we want to participate in the refinement process and make sure that our suggested sequences fare well until the end. Thank you.
How about this Ayurvedic Tava or Microbial Vita? They can tell the persisting and lot of lots of work has been done. But as this virus, no cure or something. But 90% has been killed using Tava compared to pathogen.
So any work has been done regarding that?
Yeah,
In NIMAX, they've already proved it regarding this dengue.
Yeah. So there is sparse literature on the influence of medicinal plants, Indian medicinal plants, on preventing and combating diseases, including dengue. For example, the Kapasava Kudinir was used for the SARS-CoV-2, maybe you're referring to that. And also for dengue especially, there are a couple of plants like Nilavembu, which has been talked about a lot. And there are.
But this is a common weed, ma'am, easily available.
Easily available.
Yeah.
Euphorbia,
something.
There have been some papers on the one hand to know what are the secondary metabolites, the chemicals which act like the drugs, and make sure that the dengue production is not more in the host. And but then we cannot say they have proved and they have completed, because there are so many facets to it. Because you have to do a lot more. For example, doing mapping the secondary metabolites is good, but not enough. But then getting in vitro assays to show that the dengue virus particles are not replicating is good, but that's not enough. And then we have to also show in vivo assays, maybe in a BCL3, we have to actually grow them and then show that what is happening in the cellular level. And then we also eventually have to take it forward on clinical trials, right? Because in India, people are already practicing. They are willing and they trust and they are already consuming this. But from the scientific point of view, when we do a clinical trial, we measure parameters that become so invaluable and provide that scientific validation. Maybe we should also look at that, and look at antibody production as they go through the Nilavembu treatment or whatever. So the clinical trials are pretty expensive, but we are actually open and working with some Ayurvedic hospitals, like in the city. So there's a lot more to do. And
Thank
we
you.
should, I think scientists take
Yeah,
it
yeah.
forward.
Thank
at
you,
all
ma'am.
levels.
Thank you.
Thank you.
Thank you, ma'am, for that insightful and enriching lecture. The session is now open for discussion. Any questions?
The envelope protein, you said it has three domains.
Yeah.
But the envelope protein orientation in a virus is very important. Among three domains, which one will be exposed?
Yeah, usually
Because
it's-
that is the one which is going to
Yeah.
be immediately interact with the
Correct,
antibody.
Correct. Yeah, so the viral coat protein is a very symmetric and highly stable structure because inside it, the RNA of the virus is held. So it has to be very stable, it is thermostable and all that. But it also is structurally different, and as it matures, as the virus get into the host, in influence of pH and so on, it goes through large conformational changes. That means the envelope protein itself presents outside to the host, especially the D3 domain. That's why most of the attention was given for the D3 domain. I think there was a group, a Khanna group, which had worked on the vaccine using that. So to the answer to your question is, the third domain is more solid accessible and available for the host.
So it will be exposed outside.
Yeah.
Yeah. So the antibodies, one you are using or prepared, whether it is going to label the virus to make sure phagocytes come and eat away, or whether it prevent envelope not to open. Which one is it?
Yeah, it's more of the former, because the 4E11 antibody is doing that. It is
Okay.
making sure that much of the viral production is not happening. But then what is also happening is the D2 and the D1 domains are not being naive. They are also partially exposed. You know, they are a little bit outside. That's why that loop is getting into all these side reactions. So that is why we are focusing on the D1, D2 domain, not only the D3 domain.
Because that's very important. Epitope exposure to immediately antibodies.
Correct. Correct.
That's very important.
Much of the work that we were discussing previously on the Indian medicinal plants seem to focus on on the non-structural proteins, that is the viral protease, main protease, because these small molecules that come from these plants, alkaloids and so on, act as effective inhibitors so that the viral machinery is not allowed to win over the host or propagate. So they seem to be working more at the second type of mechanism. Yeah.
Thank you.
Thank you. Due to time constraint, the students can ask the questions during the break time. So thank you, ma'am, for that insightful talk once again. So we'll have a small tea break. Kindly reassemble sharp by 11:15 into the auditorium. ಅಯ್ಯೋ ಮರ್ತೇ ಹೋಗ್ಬಿಟ್ಟೆ I request Dr. Chandrayappa sir, Head R&D, to hand over a token of appreciation to Saudamini ma'am. Sharp 11:15, students have to be back in the auditorium for the inaugural session.
No, one of the conveners at 11:15 with yoga 11:5. If I didn't have a but it's okay. Like, yeah, it's okay. Like our management. request all the students to stand up. ಎಲ್ಲರಿಗೂ ಬೆಳಗಿನ ಶುಭೋದಯ. ಕರ್ನಾಟಕ ವಿಜ್ಞಾನ ಮತ್ತು ತಂತ್ರಜ್ಞಾನ ಅಕಾಡೆಮಿ ಹಾಗೂ MS ರಾಮಯ್ಯ ಕಲಾ ವಿಜ್ಞಾನ ಮತ್ತು ವಾಣಿಜ್ಯ ಕಾಲೇಜು ಬೆಂಗಳೂರು ಇವರ ಸಹಯೋಗದೊಂದಿಗೆ ಸುಸ್ಥಿರ ಭವಿಷ್ಯಕ್ಕಾಗಿ ವಿಜ್ಞಾನ ಮತ್ತು ತಂತ್ರಜ್ಞಾನ ಎಂಬ ವಿಷಯದ ಮೇಲೆ 2 ದಿನಗಳ ಕಾಲ ಹಮ್ಮಿಕೊಂಡಿರುವ ವಿಶೇಷ ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಆಗಮಿಸಿರುವ ತಮ್ಮೆಲ್ಲರಿಗೂ ಸ್ವಾಗತವನ್ನು ಬಯಸುತ್ತಾ ರಾಮಯ್ಯ ಗೀತೆಯ ಮೂಲಕ ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಚಾಲನೆ ನೀಡಲಾಗುತ್ತಿದೆ. ತಾವೆಲ್ಲರೂ ಎದ್ದು ನಿಂತು ಗೌರವವನ್ನು ಸೂಚಿಸಬೇಕಾಗಿ ಕೋರುತ್ತೇನೆ.
ಶ್ರೀ ಗಣನಾಥುನಿ ಶಾರದ ಸತ್ಕವುಲನು ಸಹಜ ಯೋಗಿ ಮುನುಲನ್ ಸುರುಲನ್ ದೇವ ಬ್ರಾಹ್ಮಣ ಧನ್ಯುನಿ ಸಕಲಾಂತರ್ಯಾಮಿ ಹರಿಣಿ ಚಾಲ ಭಜಿಂತುಂ
ज्ञान विज्ञान विद्या विनय भूषण
ಈ ರಾಮಯ್ಯ ಸಮೂಹ ಸಂಸ್ಥಾನ
ज्ञान विज्ञान विद्या विनय भूषण ई रामय्य समूह संस्थान तारे निहारितेयागಲಿ ಈ
ಪ್ರೇರಣೆ ನಿರ್ಮಲ ಬಾಳ ಉದ್ದೀಪನೆ ಅನುದಿನ ಅನುಕ್ಷಣ ಹೊಸ ಅನ್ವೇಷಣೆ ಭಾರತದ ಸಿರಿ ಸಾಧನೆ ಶಾಂತಿ ಪ್ರೀತಿ ಸಂವರ್ಧನೆ ಕೃಷಿ ಕೈಗಾರಿಕೆ ಕಂಡು ಸಂಶೋಧನೆ ನಾಡ ಮುನ್ನಡೆಯ ನಿಶಾನೆ ನಾವೆಲ್ಲ ಒಂದೆಂಬ ಭಾವನೆ ಓಹೋ ಜ್ಞಾನವೇ ಶೋಧನೆ ಓಹೋ ಜ್ಞಾನವೇ ಸಾಧನೆ ಜ್ಞಾನ ವಿಜ್ಞಾನ ಸಂವರ್ಧನೆ ಓಹೋ ಜ್ಞಾನವೇ ರಂಜನೆ ಓಹೋ ಜ್ಞಾನವೇ ಪ್ರೇರಣೆ ನಿರ್ಮಲ ಬಾಳ ಉದ್ದೀಪನೆ
ವೇದಿಕೆಯ ಮೇಲೆ ಉಪಸ್ಥಿತರಿರುವ ಗಣ್ಯರನ್ನು ಸ್ವಾಗತಿಸಲಿದ್ದಾರೆ ಅಕಾಡೆಮಿಯ ಮಾನ್ಯ ಅಧ್ಯಕ್ಷರು ಮತ್ತು ತುಮಕೂರು ವಿಶ್ವವಿದ್ಯಾಲಯ ವಿಶ್ರಾಂತ ಕುಲಪತಿಗಳಾದ ಡಾಕ್ಟರ್ ರಾಜಾಸಾಬ್ ಎಚ್ ರವರು.
ವೇದಿಕೆಯ ಮೇಲೆ ಆಸೀನರಾಗಿರುವಂತಹ ಮಾನ್ಯರಾದ ಡಾಕ್ಟರ್ MR ಸೀತಾರಾಮ್ ಸರ್ ಅವರೇ, ಡಾಕ್ಟರ್ ಕರಿಸಿದ್ದಪ್ಪ ಅವರೇ, ಡಾಕ್ಟರ್ ಎಚ್ ವಿ ಪಾರ್ಶ್ವನಾಥ್ ಅವರೇ, ಶ್ರೀ ರಾಮಚಂದ್ರ ಅವರೇ, ಪ್ರಿನ್ಸಿಪಾಲರಾದಂಥ ಶ್ರೀಮತಿ ಡಾಕ್ಟರ್ ಪುಷ್ಪ ಅವರೇ, ನಮ್ಮ ಅಕಾಡೆಮಿಯ CEO ಆದಂಥ ಡಾಕ್ಟರ್ ಆನಂದ್ ಅವರೇ, ಸಭೆಯಲ್ಲಿ ಉಪಸ್ಥಿತಿರುವಂತಹ ಮಾನ್ಯರಾದ ನಮ್ಮ KSTA ಅಕಾಡೆಮಿಯ ಗೌರವಾನ್ವಿತ ಸದಸ್ಯರುಗಳಾದಂತಹ ಡಾಕ್ಟರ್ ಸಂಗೂರು ಮಠ್ ಅವರೇ, ಡಾಕ್ಟರ್ ಚಂದ್ರಶೇಖರ ಅವರೇ, ಹಾಗೂ ಸೈನ್ಸ್ ಆ್ಯಂಡ್ ಟೆಕ್ನಾಲಜಿ ಫಾರ್ ಸಸ್ಟೈನಬಲ್ ಫ್ಯೂಚರ್ ಈ ಉಪನ್ಯಾಸ ಮಾಲಿಕೆಯಲ್ಲಿ ಭಾಗವಹಿಸಲು ಆಗಮಿಸಿರುವ ಎಲ್ಲ ನಮ್ಮ ಪ್ರೀತಿಯ ವಿದ್ಯಾರ್ಥಿ ಮತ್ತು ವಿದ್ಯಾರ್ಥಿನಿಯರೇ, ಮಾಧ್ಯಮ ಮಿತ್ರರೇ, KSTA ಮತ್ತು ರಾಮಯ್ಯ ಕಾಲೇಜು ಮತ್ತು ಇತರ ಕಾಲೇಜಿನ ಎಲ್ಲ ಅಧ್ಯಾಪಕ ಮತ್ತು ಅಧ್ಯಾಪಕೇತರ ಸ್ನೇಹಿತರೆ, ತಮ್ಮೆಲ್ಲರಿಗೂ ಹಾರ್ದಿಕ ಸ್ವಾಗತವನ್ನು ಕೋರಲು ನಮಗೆ ಸಂತೋಷವಾಗುತ್ತಿದೆ. ಈ ಸುಸಂದರ್ಭದಲ್ಲಿ ಗೌರವಾನ್ವಿತ ಮುಖ್ಯ ಅತಿಥಿಗಳಾಗಿ ಆಗಮಿಸಿರುವಂತಹ ಡಾಕ್ಟರ್ MR ಸೀತಾರಾಮ್ ಸರ್ ಅವರ ಕಿರುಚು ಪರಿಚಯ ಮಾಡಿಕೊಡಲು ನಮಗೆ ಹೆಮ್ಮೆ ಎನಿಸುತ್ತಿದೆ. ತಮಗೆಲ್ಲರಿಗೂ ಗೊತ್ತಿರುವ ಹಾಗೆ ಕರ್ನಾಟಕದ ವಿಧಾನಸೌಧದಲ್ಲಿ ವಿಧಾನಸಭೆ ಮತ್ತು ವಿಧಾನ ಪರಿಷತ್ತಿನ ಕಾರ್ಯಕಲಾಪಗಳು ನಡೀತಾ ಇದೆ, ಇವತ್ತು ನಡೀತಾ ಇದೆ. ನಾವು ಈ ಹಿಂದೆ ಡಾಕ್ಟರ್ ಸೀತಾರಾಮ್ ಸರ್ ಅವರನ್ನು ಕೇಳಿಕೊಂಡಾಗ ಅವರು ಒಪ್ಕೊಂಡಿದ್ದರು. ಆನಂತರ ಈ ಕಾರ್ಯಕಲಾಪಗಳು ಸ್ಟಾರ್ಟ್ ಆದಮೇಲೆ ನಮಗೆ ಸ್ವಲ್ಪ ಆತಂಕ ಆಯಿತು. ವಿಧಾನ ಪರಿಷತ್ತಿನ ಕಲಾಪ ನಡೀತಾ ಇದೆ, ಸಾಹೇಬರು ಬರ್ತಾರೋ ಇಲ್ಲೋ ಅಂತ. ಬಹಳ ಅಳುಕಿನಿಂದ ಆತಂಕದಿಂದ ಮೊನ್ನೆ ಅವರನ್ನು ನಾವು ಹೋಗಿ ಭೇಟಿ ಮಾಡಿದಾಗ ನಾನು ಮತ್ತು ಡಾಕ್ಟರ್ ಪುಷ್ಪ ಮೇಡಮ್ ಅವರು ಒಂದೇ ಮಾತಿನಲ್ಲಿ ಅವರು ಹೇಳಿದರು, ನಾನು ಬರ್ತೀನಿ, ಬಂದು ಒಂದು ಸ್ವಲ್ಪ ಹೊತ್ತು ಮಾತಾಡಿ ನಾನು ವಿಧಾನ ಪರಿಷತ್ ಕಲಾಪಕ್ಕೆ ಹೋಗ್ತೀನಿ, ಜಾಸ್ತಿ ಸಮಯ ನನಗೆ ಕೊಡೋಕ್ಕಾಗಲ್ಲ, ಆದರೆ ಬೇಗ ನನಗೆ ಕಳಿಸ್ಕೊಟ್ಬಿಡಿ ಅಂದರು. ಆವಾಗ ನಮಗೆ ಇಷ್ಟು ಸಂತೋಷ ಆಯಿತು, ಎಲ್ಲ ಟೆನ್ಷನ್ನು ಅವತ್ತಿಗೆ ಮುಗಿದೋಗ್ಬಿಡ್ತು. ಯಾಕೆಂದರೆ ಹೇಗೆ ಈ ಪ್ರೋಗ್ರಾಮನ್ನು ಸ್ಟಾರ್ಟ್ ಮಾಡೋದು ಸಾಹೇಬ್ರಿಲ್ದೆ ಅನ್ನೋ ಮನಸ್ಸಿಗೆ ಒಂದು ದುಗುಡ ಆಗಿತ್ತು. ಅಂತಹ ಗುರುತರವಾದ ಕಾರ್ಯವನ್ನು ಬದಿಗೊತ್ತಿ ಮೊದಲಿಗೆ ಬಂದಿರೋದು ನಮ್ಮ ಕಾರ್ಯಕ್ರಮಕ್ಕೆ. ಇದಾದ ನಂತರ ಅವರು ಕಲಾಪಕ್ಕೆ ವಿಧಾನ ಪರಿಷತ್ತಿನ ಕಾರ್ಯಕಲಾಪದಲ್ಲಿ ಭಾಗವಹಿಸ್ತಾರೆ. ಅವರು ದೊಡ್ಡ ಮನಸ್ಸು ಮಾಡಿ ಇವತ್ತು ಇಲ್ಲಿಗೆ ಬಂದಿದ್ದಾರಲ್ಲ, ಆದ್ದರಿಂದ ಅದು ನಮ್ಮ ಸೌಭಾಗ್ಯ ಅಂತ ನಾನು ಭಾವಿಸ್ತೀನಿ. ಮಾನ್ಯರು 2016ರಿಂದ 2018ರ ಅವಧಿಯಲ್ಲಿ ಕರ್ನಾಟಕ ಸರ್ಕಾರದ ಯೋಜನೆ, ಸಾಂಖ್ಯಿಕ ವಿಜ್ಞಾನ ಮತ್ತು ತಂತ್ರಜ್ಞಾನ ಇಲಾಖೆಯ ಗೌರವಾನ್ವಿತ ಮಂತ್ರಿಗಳಾಗಿದ್ದರು. ಈಗೇನು ನಮ್ಮ ವಿಜ್ಞಾನ ತಂತ್ರಜ್ಞಾನ ಅಕಾಡೆಮಿ ಇದೆ, ಈ ಕ್ಯಾಂಪಸ್ ಇದೆ, ಈ ಬಿಲ್ಡಿಂಗ್ ಇದೆ, ಆ ಗೆಸ್ಟ್ ಹೌಸ್ ಇದೆ, ಏನೆಲ್ಲ ಪರಿಸರ ಇಲ್ಲಿ ಸುಂದರವಾಗಿದೆ, ಇದಕ್ಕೆಲ್ಲ ಸಹಾಯ ಮಾಡಿದಂಥವರು ಆಗಿನ ಸಮಯದಲ್ಲಿ ಮಿನಿಸ್ಟ್ರ್ ಆಗಿದ್ದಂತಹ ನಮ್ಮ ಸೀತಾರಾಮ್ ಸರ್ ಅವರು. ಆದ್ದರಿಂದ ನನ್ನ ಮನಸ್ಸಿನಲ್ಲಿ ಯಾವಾಗೂ ಅನ್ನಿಸ್ತಾ ಇತ್ತು, ಒಂದು ಸಲ ಸೀತಾರಾಮ್ ಸರ್ ಅವ್ರನ್ನ ಕರ್ಕೊಂಡು ಬಂದು ನಮ್ಮ ಕ್ಯಾಂಪಸ್ ತೋರಿಸ್ಬೇಕು ಅಂತ. ಇವತ್ತು ಆ ಕನಸು ನನಸಾಗಿದೆ. ಆದ್ದರಿಂದ ವೈಯಕ್ತಿಕವಾಗಿಯೂ ಸಹ ಅವರಿಗೆ ನಾನು ಧನ್ಯವಾದಗಳನ್ನು ಸಲ್ಲಿಸ್ತೇನೆ. ಈಗ ಕರ್ನಾಟಕ ವಿಧಾನ ಪರಿಷತ್ತಿನ MLC ಸಹ ಅವರು ಆಗಿದ್ದಾರೆ. ಅದರ ಜೊತೆಗೆ ಅಧ್ಯಕ್ಷರು, ಕರ್ನಾಟಕ ಗೋಕುಲ ಎಜುಕೇಷನ್ ಫೌಂಡೇಶನ್ ಅಧ್ಯಕ್ಷರಾಗಿ ಶಿಕ್ಷಣ ರಂಗದಲ್ಲಿ ಅಮೂಲ್ಯ ಸೇವೆಯನ್ನು ಸಲ್ಲಿಸುತ್ತಿದ್ದಾರೆ. ಸಾವಿರಾರು ವಿದ್ಯಾರ್ಥಿಗಳಿಗೆ ಶಿಕ್ಷಣ ಸೌಲಭ್ಯ ಕಲ್ಪಿಸಿಕೊಟ್ಟಿದ್ದಾರೆ. ರಾಮಯ್ಯ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆಗಳು ಇಡೀ ನಾಡಿನಲ್ಲಿ ದೇಶ ವಿದೇಶಗಳಲ್ಲಿ ಮೌಲ್ಯಯುತ ಶಿಕ್ಷಣ ನೀಡುವಲ್ಲಿ ಹೆಸರುವಾಸಿಯಾಗಿದೆ. ತಮಗೆಲ್ಲ ಅದು ಗೊತ್ತಿದೆ. ಅದೇ ರೀತಿ ಸೈನ್ಸ್ ಸೆಂಟರ್ ವಿಜ್ಞಾನ ಕೇಂದ್ರಗಳನ್ನು ಧಾರವಾಡದಲ್ಲಿ, ಮಂಗಳೂರಿನಲ್ಲಿ ಇರುವಂಥ ವಿಜ್ಞಾನ ಕೇಂದ್ರಗಳನ್ನು ಉನ್ನತೀಕರಿಸಿ ಈಗ ರಾಜ್ಯದಲ್ಲಿ ಅತ್ಯುತ್ತಮ ವಿಜ್ಞಾನ ಕೇಂದ್ರಗಳಾಗಿ ಅವು ಕೆಲಸ ಮಾಡ್ತಾ ಇದ್ದಾವೆ. ಅದ್ರ ಜೊತೆಗೆ ಇನ್ನೂ ಉಳಿದ ಡಿಸ್ಟ್ರಿಕ್ಟ್ಗಳಲ್ಲಿ 22 ಉಪಪ್ರಾದೇಶಿಕ ವಿಜ್ಞಾನ ಕೇಂದ್ರಗಳನ್ನು ಅವರ ಸಮಯದಲ್ಲಿ ಅವರು ಪ್ರಾರಂಭ ಮಾಡಿದರು. ಮಂಗಳೂರಿನ ಪಿಳ್ಳಿಕುಳ ಅಂತ ತಾವು ಕೇಳಿರ್ತೀರ, ಅಲ್ಲಿ 3D ಪ್ರೊಜೆಕ್ಷನ್ ಫೆಸಿಲಿಟಿ ಇರುವಂತಹ, ಇಲ್ಲಿ ನಮ್ಮ ನೆಹರು ಪ್ಲಾನೆಟೋರಿಯಂ ಏನು ಬೆಂಗಳೂರಲ್ಲಿದೆ, ಆ ರೀತಿಯಂತಹ ಫೆಸಿಲಿಟಿಯನ್ನು ಮಂಗಳೂರಿನಲ್ಲಿ ಅವರು ನಿರ್ಮಿಸಿಕೊಟ್ಟಿದ್ದಾರೆ. ಅದೇ ರೀತಿ ಮಿನಿ ತಾರಾಲಯಗಳ ನಿರ್ಮಾಣವನ್ನು ಮಾಡಿದ್ದಾರೆ. ಸುಮಾರು ಬೇರೆ ಬೇರೆ ಡಿಸ್ಟ್ರಿಕ್ಟ್ಗಳಲ್ಲಿ ಬಾಗಲಕೋಟೆ, ವಿಜಯಪುರ, ಶಿವಮೊಗ್ಗ, ಮಡಿಕೇರಿ, ಬೆಳಗಾವಿ, ಗದಗ ಈ ಜಿಲ್ಲೆಗಳಲ್ಲಿ ಮಿನಿ ತಾರಾಲಯಗಳು ಸಹ ಈ ಕೆಲಸ ಮಾಡ್ತಿದ್ದಾವೆ. ಈ ಒಂದು ಇದರ ಕ್ರೆಡಿಟ್ ಶ್ರೇಯಸ್ಸು ಮಾನ್ಯ ಸೀತಾರಾಮ್ ಸರ್ ಅವರಿಗೆ ಹೋಗಬೇಕು. ಆಮೇಲೆ ಅವರು ಮಿನಿಸ್ಟ್ರ್ ಇದ್ದ ಸಮಯದಲ್ಲಿ ಶಾಲೆಯ ಅಂಗಳದಲ್ಲೇ ತಾರಾಲಯ ಇರಬೇಕು ಅನ್ನೋ ಒಂದು ಕಾನ್ಸೆಪ್ಟನ್ನು ಹುಟ್ಟು ಹಾಕಿದವರು. ಹಾಗಾಗಿ ವಿನೂತನ ಯೋಜನೆಯನ್ನು ಅವರು ಅನುಷ್ಠಾನಕ್ಕೆ ತಂದಿದ್ದಾರೆ. ಇದು ಸುಮಾರು 5 ಡಿಸ್ಟ್ರಿಕ್ಟ್ನಲ್ಲಿ ಈಗ ನಡೀತಾ ಇದೆ. ಇದು ಅಲ್ಲದೆ ಐರ್ಲೆಂಡ್ ಅನ್ನೋ ಮಾದರಿ ಸೈನ್ಸ್ ಗ್ಯಾಲರಿಯನ್ನು ನೀವು ವೆಟರ್ನರಿ ಸೈನ್ಸ್ ಕಾಲೇಜು ಹೋಗ್ತಿರುವಾಗ ಹೈವೇನಲ್ಲೇ ಬಲಗಡೆಗೆ ಕಾಣಿಸುತ್ತೆ ಸೈನ್ಸ್ ಗ್ಯಾಲರಿ ಅಂತ. ಅದು ಇಡೀ ವಿಶ್ವದಲ್ಲಿ ಸ್ಟ್ಯಾಂಡರ್ಡ್ ಆಗಿರುವಂಥ ಸೈನ್ಸ್ ಗ್ಯಾಲರಿ. ಅವರು ಮಿನಿಸ್ಟರ್ ಆದಂಥ ಸಮಯದಲ್ಲಿ ಅದರ ಹುಟ್ಟು ಹಾಕಿದರು. ಮೊನ್ನೆ ಈ ವರ್ಷದಲ್ಲಿ ಸೈನ್ಸ್ ಗ್ಯಾಲರಿಗೇ ಕರ್ನಾಟಕ ಸರ್ಕಾರ 10 ಕೋಟಿ ರೂಪಾಯಿ ಅನುದಾನವನ್ನು ಸಹ ನೀಡಿದೆ. ಆದ್ದರಿಂದ ಕರ್ನಾಟಕ ಸರ್ಕಾರಕ್ಕೆ ನಾವು ಅಭಿನಂದನೆಗಳನ್ನು ಸಲ್ಲಿಸ್ತೀವಿ. ಅದೇ ರೀತಿ KSTಗೆ ನಾನು ಹೇಳಿದ ಹಾಗೆ ಸಾಕಷ್ಟು ಅನುದಾನವನ್ನು ಅವ್ರ ಸಮಯದಲ್ಲಿ ಒದಗಿಸಿದ್ರು. ಅದನ್ನು ನಾನು ಬಹಳ ಹೃತ್ಪೂರ್ವಕವಾಗಿ ನೆನೆಸ್ಕೊಳ್ತೇನೆ. ಯಾಕೆಂದರೆ ಆವಾಗ ನಾನು ವೈಸ್ ಚಾನ್ಸಲರ್ ಇದ್ದೆ ತುಮಕೂರು ಯೂನಿವರ್ಸಿಟಿ. ಸಾಹೇಬ್ರ ಪರಿಚಯ ನನಗಿರಲಿಲ್ಲ. ಒಂದು ಫಂಕ್ಷನ್ ಇತ್ತು ಮಧುಗಿರಿನಲ್ಲಿ. ಅಲ್ಲಿ ನಮ್ಮ ಚೀಫ್ ಮಿನಿಸ್ಟರ್ ಸಿದ್ದರಾಮಯ್ಯ ಸರ್ ಬರ್ತಾಯಿದ್ರು. ನಾನು ಶಾಲಿನಿ ಅವರು ಡಿಸ್ಟ್ರಿಕ್ಟ್ ಇನ್ಚಾರ್ಜ್ ಇದ್ದರು ಸರ್. ನಾನು ಅವರು ಇಬ್ಬರು ಅವರೊಂದು ಬುಕೆ ನಾನೊಂದು ಬುಕೆ ಹಿಡ್ಕೊಂಡು ಚೀಫ್ ಮಿನಿಸ್ಟ್ರಿಗೆ ಕೊಡಬೇಕು ಅಂತ ನಿಂತ್ಕೊಂಡಿದ್ವಿ. ಅದು ಹೆಲಿಕಾಪ್ಟರ್ ಬಂದು ಇಳೀತು. ಅದ್ರ ಜೊತೆಗೆ ನಮ್ಮ ರಾಜಣ್ಣ ಅವರು ಇಳಿದ್ರು. ರಾಜಣ್ಣ ಅಲ್ಲ, ನಮ್ಮ ಜಯಚಂದ್ರ ಸರ್ ಅವರು ಇಳಿದ್ರು. ಆ ಜೊತೆಗೆ ಸಾಹೇಬರು ಅವ್ರ ಜೊತೆಗಿದ್ದರು. ನನಗೆ ಹೆಂಗಪ್ಪ ಮಾಡೋದು, ನಾನು ಬರೀ ಒಂದು ಬುಕ್ಕೆ ತಂದಿದ್ದೀನಿ. ಚೀಫ್ ಮಿನಿಸ್ಟ್ರಿಗೆ ಆ ಬುಕ್ಕೆ ಕೊಟ್ವಿ. ಇನ್ನೊಂದು ಬುಕ್ಕೆ ಕೊಡೋಕೆ ನಮ್ಮ ಕೈನಲ್ಲಿರಲಿಲ್ಲ. ನನ್ನ ಮನಸ್ಸಿಗೆ ಒಂಥರ ಆಗ್ತಾಯಿತ್ತು. ನಮಸ್ತೆ ಸರ್ ಅಂತ ಹೇಳಿದೆ. ಓ, ನೀವು ರಾಜಾ ಸಾಬ್ ಅಲ್ಲ, ನೀವು ನನಗೆ ಗೊತ್ತು ಅಂತ ಹೇಳಿದರು. ನನಗೆ ಅವತ್ತು ಒಂದು ರೀತಿಯ ಹೇಗಾಯಿತು ಅಂದರೆ ನಾನು 9ನೇ ಆಕಾಶಕ್ಕೆ ಹೋಗ್ಬಿಟ್ಟಿದ್ದೀನೇನೋ ಅನ್ನಿಸ್ತು. ಅಷ್ಟು ಅವರು ನೆನ್ಪಿಟ್ಕೊಂಡಿದ್ರು. ನಿಮಗೆ ಒಂದು ವಿಜ್ಞಾನ ಕೇಂದ್ರವನ್ನು ಕೊಡೋದಿದೆ ತುಮಕೂರಿಗೆ, ಅದು ನೆನೆಗುದಿಗೆ ಬಿದ್ದಿದೆ, ನೀವು ಜಾಗ ಕೊಟ್ಟಿದ್ದೀರಿ ಅಂತೇಳಿ. ಸರ್ ಹೌದು ಸರ್, ನೀವು 4 ಕೋಟಿ ರೂಪಾಯಿನೂ ನಮಗೆ ಕೊಟ್ಟಿದ್ದೀರಿ, ನಾವು 2 ಎಕರೆ ಜಾಗ ತುಮಕೂರಲ್ಲಿ ಕೊಟ್ಟಿದ್ದೀವಿ ಅಂತ ಹೇಳಿದೆ. ಭಾಳ ಸಂತೋಷಪಟ್ಟರು. ಆಮೇಲೆ ಹೋಗುವಾಗ ಹೇಳಿದರು, ನೀವೊಂದು ಅಪ್ಲಿಕೇಶನ್ ಕಳಿಸ್ಕೊಡಿ, ನಾವು 8 ಕೋಟಿ ನಿಮಗೆ ಕೊಡ್ತೀವಿ ಅಂತ ಹೇಳಿದರು. ಅವತ್ತು ಸ್ಯಾಟರ್ಡೇ ಇತ್ತು. ಮತ್ತು ಸೋಮವಾರದ ದಿನವೇ ನಾನು ನಮ್ಮ ರೆಪ್ರೆಸೆಂಟೇಟಿವ್ಗಳಿಗೆ ಕಳಿಸಿ 8 ಕೋಟಿ ಪ್ರಪೋಸಲ್ ಕೊಟ್ಟೆ. ಆ 8 ಕೋಟಿ ಪ್ರಪೋಸ್ನಲ್ಲಿ ಇನ್ನೂ ಒಳ್ಳೆಯ ಕೆಲಸ ಆಯಿತು. ತುಮಕೂರು ವಿಶ್ವವಿದ್ಯಾಲಯಕ್ಕೆ 250 ಎಕರೆ ಭೂಮಿ ಏನು ಸಿಕ್ತಲ್ಲ, ಅದರಲ್ಲಿ ನಾವು ಈಗ 4 ಎಕರೆ ಜಮೀನನ್ನು ಸೈನ್ಸ್ ಸೆಂಟ್ರಿಗೆ ಕೊಟ್ಟಿದ್ದೀವಿ. 8 ಎಕರೆ ಗೌರ್ಮೆಂಟ್ನಿಂದ 8 ಕೋಟಿ ಗೌರ್ಮೆಂಟಿಂದ ಬಂದಿದೆ. ಒಂದು ಸಮಯದಲ್ಲಿ ಒಳ್ಳೆಯ ಸಮಯದಲ್ಲಿ ಒಳ್ಳೆಯವರು ಆಡಿದ ಮಾತುಗಳೇ ಒಂದು ರೀತಿ ದೇವತೆಗಳು ಆಡಿದಂಥ ಮಾತುಗಳು ಅಂತ ನನಗನ್ನಿಸ್ತಿದೆ. ಅವತ್ತು ಅವರು 8 ಕೋಟಿನೂ ಕೊಟ್ಟರು. ಭಾಳ ವರ್ಷಗಳಿಂದ ತುಮಕೂರಲ್ಲಿ ಭಾಳ ಅದು ಕಾಂಟ್ರವರ್ಷಿಯಲ್ ಆಗಿಬಿಟ್ಟಿತ್ತು ಸೈನ್ಸ್ ಸೆಂಟರು. ಸಾಹೇಬರು ಬಂದ ಮೇಲೆ ಅದನ್ನು ಸೆಟಲ್ ಮಾಡಿಬಿಟ್ರು, ಹಣವನ್ನು ಕೊಟ್ಟರು. ಈಗ ಅದು ತುಮಕೂರಿನ PG ಮೇನ್ ಕ್ಯಾಂಪಸ್ ಹೊರಗಡೆ ಅಲ್ಲಿ ಆಗಿದೆ. ಅಲ್ಲಿ ಸೈನ್ಸ್ ಸೆಂಟರು ಸಹ ಬರ್ತಾಯಿದೆ. ಹೀಗೆ ಅವರನ್ನು ನೆನೆಸ್ಕೊಳ್ತಾ ಹೋದರೆ ಸಮಯ ಭಾಳ ಕಡಿಮೆ ಇದೆ. ಅವರು ಇಡೀ ದೇಶದಲ್ಲಿ, ಇಡೀ ನಮ್ಮ ನಾಡಿನಲ್ಲಿ ಅತ್ಯುತ್ತಮವಾದ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆಗಳನ್ನು ಮಾಡಿದ್ದಾರೆ, ಹುಟ್ಟು ಹಾಕಿದ್ದಾರೆ. ಎಷ್ಟು ಉತ್ತಮವಾದ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆಗಳು, ಅದು RIT ಇರ್ಬೋದು, ರಾಮಯ್ಯ ಇನ್ಸ್ಟಿಟ್ಯೂಟ್ ಆಫ್ ಟೆಕ್ನಾಲಜಿ, ರಾಮಯ್ಯ ಇನ್ಸ್ಟಿಟ್ಯೂಟ್ ಆಫ್ ಮ್ಯಾನೇಜ್ಮೆಂಟು, ರಾಮಯ್ಯ ಸೈನ್ಸ್ ಕಾಲೇಜು, ರಾಮಯ್ಯ ಮೆಡಿಕಲ್ ಕಾಲೇಜು, ರಾಮಯ್ಯ ಯೂನಿವರ್ಸಿಟಿ, ಹೀಗೆ ನಾನು ಒಂದು ದೊಡ್ಡ ಲಿಸ್ಟನ್ನೇ ಮಾಡ್ಬೋದು. ಇಷ್ಟು ಕೆಲಸವನ್ನು ಒಂದು ಫ್ಯಾಮಿಲಿಯಿಂದ ಕೆಲವೇ ಬ್ರದರ್ಸ್ನಿಂದ ಮಾಡೋಕ್ಕೆ ಸಾಧ್ಯ ಅಂತ ನನಗೂ ಊಹೆ ಮಾಡ್ಕೊಳ್ಳೋಕ್ಕೂ ಆಗೋದಿಲ್ಲ. ಅಂತಹ ಒಂದು ದೊಡ್ಡ ಪರ್ಸನಾಲಿಟಿ ಅವರು. ಅವರ ಸೇವೆಯನ್ನು ಗಮನಿಸಿ ಮೈಸೂರು ವಿಶ್ವವಿದ್ಯಾಲಯ ಹೋದ ವರ್ಷ ಗೌರವ ಡಾಕ್ಟ್ರೇಟನ್ನು ಕೊಟ್ಟಿದೆ. ಅದು ಭಾಳ ಅಭಿಮಾನದ ಸಂಗತಿ. ಹಿ ವಾಸ್ ಹಾನರ್ಡ್ ವಿತ್ ದಿ ಡಾಕ್ಟರಲ್ ಹಾನರರಿ ಡಾಕ್ಟ್ರೇಟ್ ಫ್ರಮ್ ಯೂನಿವರ್ಸಿಟಿ ಆಫ್ ಮೈಸೂರು. ಆ ಪದವಿಯಿಂದ ಸಾಹೇಬರ ಒಂದು ಗೌರವ ಹೆಚ್ಚಾಗಿದೆ ಅನ್ನೋದು ಅಷ್ಟೇ ಅಲ್ಲ, ಯೂನಿವರ್ಸಿಟಿ ಗೌರವನೂ ಹೆಚ್ಚಾಗಿದೆ, ನಮ್ಮ ರಾಮಯ್ಯ ಗ್ರೂಪ್ ಆಫ್ ಇನ್ಸ್ಟಿಟ್ಯೂಷನ್ನ ಗೌರವನೂ ಹೆಚ್ಚಾಗಿದೆ. ಅಂಥ ಒಂದು ಒಳ್ಳೆಯ ಪರ್ಸನಾಲಿಟಿ ಅವರು. ಆಮೇಲೆ ನಾನು ಇತ್ತೀಚಿನ ಒಂದು 3-4 ವರ್ಷಗಳಿಂದ ಅವರನ್ನು ತುಂಬ ಹತ್ತಿರದಿಂದ ನೋಡ್ತಾ ಇದ್ದೀನಿ. ಅವರ ಸಾಮಾಜಿಕ ಕಳಕಳಿ, ಅವರ ರಾಜಕೀಯ ನಿಲುವುಗಳು, ಆಮೇಲೆ ಯಾರಿಗೆ ಅವಶ್ಯಕತೆ ಇದೆ ಈ ಶಿಕ್ಷಣ, ಅವರಿಗೆ ಶಿಕ್ಷಣವನ್ನು ಒದಗಿಸಬೇಕು, ಬೇರೆಯವರನ್ನು ನಾವು ಮಾನವೀಯ ಧರ್ಮದಿಂದ ನೋಡಬೇಕು, ಆಲ್ ಆರ್ ಹ್ಯೂಮನ್ ಬೀಯಿಂಗ್ಸ್, ಕಂಪ್ಯಾಷನೇಟ್ ಆಗಿರಬೇಕು, ಕಷ್ಟಪಟ್ಟವ್ರಿಗೆ ಕಷ್ಟಪಡ್ತಾ ಇರೋರಿಗೆ ಹೆಲ್ಪ್ ಮಾಡಬೇಕು, ಸುತ್ತಮುತ್ತ ಇರೋರಿಗೆ ಸಹಾಯ ಮಾಡಬೇಕು. ಈ ರೀತಿಯ ಒಂದು ಅವರ ವೈಚಾರಿಕ ಮನೋವೃತ್ತಿ, ಸಾಮಾಜಿಕ ಕಳಕಳಿ ಇದೆಯಲ್ಲ, ಇದು ಎಲ್ಲರಿಗೂ ಇರೋದಿಲ್ಲ. ಅವ್ರಿಗೆ ದೇವರು ಎಲ್ಲದನ್ನೂ ಕೊಟ್ಟಿದ್ದಾನೆ. ಆದರೂ ಅವರು ಸಮಾಜದ ಬಗ್ಗೆ ಆಲ್ವೇಸ್ ಯಾವಾಗಲೂ ಒಳಿತನ್ನೇ ಬಯಸ್ತಿದ್ದಾರೆ. ಶಿಕ್ಷಣವನ್ನು ನಮ್ಮ ಸಮಾಜದ ವಿದ್ಯಾರ್ಥಿಗಳಿಗೆ, ಬಡ ವಿದ್ಯಾರ್ಥಿಗಳಿಗೆ, ಮಿಡಲ್ ಕ್ಲಾಸ್ ವಿದ್ಯಾರ್ಥಿಗಳಿಗೆ ಶಿಕ್ಷಣ ಕೊಡ್ತಾ ಇದ್ದಾರೆ. ಶಿಕ್ಷಣ ಕೊಡೋದು ಅಂದರೆ ಇಡೀ ದೇಶ ಕಟ್ಟೋದು ಅಂತ ನಾನು ಭಾವಿಸಿದ್ದೀನಿ. ಒಳ್ಳೆಯ ಶಿಕ್ಷಣ ಕೊಟ್ಟರೆ ಒಳ್ಳೆಯ ದೇಶವನ್ನು ಕಟ್ಬೋದು. ಆ ವಿದ್ಯಾರ್ಥಿಗಳಲ್ಲಿ ಒಳ್ಳೆಯ ಮಾನವೀಯ ಮನೋಭಾವಗಳನ್ನು ತುಂಬಿದರೆ ದೇಶ ನಾಳೆ ಒಳ್ಳೆಯ ದೇಶ ಆಗಲಿಕ್ಕೆ ನಿಸ್ಸಂಶಯವಾಗಿ ಆಗುತ್ತೆ. ಇಂತಹ ಒಳ್ಳೆಯ ವ್ಯಕ್ತಿತ್ವ ಇರೋರು, ಸಾಧನೆ ಮಾಡಿರೋರು, ಸಮಾಜದ ಸೇವೆ ಮಾಡ್ತಾ ಇರೋರು ಇವತ್ತು ನಮ್ಮ ಅಕಾಡೆಮಿಗೆ ಬಂದು, ಅವ್ರದ್ದೇ ಅಕಾಡೆಮಿ ಅದು, ನಮ್ಮ ಅಕಾಡೆಮಿ ಅಲ್ಲ, ಅವ್ರದ್ದೇ ಅಕಾಡೆಮಿ, ಅವರು ಸಹಾಯ ಮಾಡಿದ್ದಾರೆ. ಇವತ್ತು ಸ್ವಲ್ಪ ಸಮಯದ ಮಟ್ಟಿಗಾದರೂ ಬಂದು ಶೋಭೆಯನ್ನು ತಂದಿದ್ದಾರೆ. ಆದ್ದರಿಂದ ಅಕಾಡೆಮಿ ವತಿಯಿಂದ, ರಾಮಯ್ಯ ಕಾಲೇಜ್ ಆಫ್ ಸೈನ್ಸ್, ಆರ್ಟ್ಸ್ ಆ್ಯಂಡ್ ಕಾಮರ್ಸ್ ಇದ್ರ ವತಿಯಿಂದ, ತಮ್ಮೆಲ್ಲರ ವತಿಯಿಂದ ಅವರನ್ನು ಹಾರ್ದಿಕವಾಗಿ ಸ್ವಾಗತಿಸ್ತೇನೆ. ನಮಸ್ಕಾರ.
ಎಲ್ಲರನ್ನೂ ಸ್ವಾಗತಿಸಿದ ಡಾಕ್ಟರ್ ರಾಜಾಸಾಬ್ ಎಚ್ ರವರಿಗೂ ಸ್ವಾಗತವನ್ನು ಬಯಸುತ್ತಾ ಧನ್ಯವಾದಗಳನ್ನು ಅರ್ಪಿಸುತ್ತೇನೆ.
2 ದಿನದ ಉಪನ್ಯಾಸ ಮಾಲಿಕೆಯ ಉದ್ದೇಶ ಮತ್ತು ರೂಪರೇಷೆಯನ್ನು ಪ್ರಸ್ತುತಪಡಿಸಲಿದ್ದಾರೆ MS ರಾಮಯ್ಯ ಕಲಾ, ವಿಜ್ಞಾನ ಮತ್ತು ವಾಣಿಜ್ಯ ಕಾಲೇಜಿನ ಪ್ರಾಂಶುಪಾಲರಾದ ಡಾಕ್ಟರ್ ಪುಷ್ಪಾ ಎಚ್ ರವರು.
Good morning, everyone. Honorable dignitaries on the dais, respected speakers, faculty members, dear students and invitees. It gives me immense pleasure to present an overview of the 2-day special lecture series on Science and Technology for Sustainable Future. This academically enriching program is jointly organized by MS Ramaya College of Arts, Science and Commerce in association with Karnataka Science and Technology Academy, Government of Karnataka. This 2-day lecture series, scheduled on 4th and 5th of February 2026, aims to explore students, researchers and faculty members to recent trends, cutting-edge research and interdisciplinary applications of science and technology. We are privileged to host an eminent panel of speakers from premier national and international institutions. As a part of pre-inaugural academic session, the lecture series began with an insightful address by Professor R. Sodamini, Professor of Biochemistry and Bioinformatics at the National Center of Biological Science, Tata Institute of Fundamental Research, Bangalore, on computational approaches to protein science. Her lecture highlighted the growing significance of bioinformatics and computational tools in advancing contemporary biological research. After the inauguration, we have the privilege of listening to Dr. Chandrasekhar M. Biradar, Earth Science Scientist and Lead, Global Growth, founder and CDM of Global Green Growth COP, Singapore. Who will be delivering a lecture on augmented science, technology and academia for restoring regenerative future, emphasizing the integration of science, technology and policy for sustainable development. We are honored to have Professor Fakhruddin B., Director of Research at University of Horticultural Science, Bagalkot, who will address the topic Genome Editing and Sustainable Agriculture, Role of CRISPR in Food Security, underlining the role of advanced biotechnological intervention in ensuring sustainable agriculture and global food security. On the second day, the lecture series will feature with Dr. K. Subhash, Scientist C at National Institute of Virology, ICMR NIV, Bengaluru Unit, who will deliberate on emerging infectious diseases, viral threats, and preparedness for the future, a topic of immense relevance in the post-pandemic era. This will be followed by Dr. Meenakshi Kamaraj, DST Inspire Faculty, Department of Bioengineering, Indian Institute of Science, Bangalore, who will present a lecture on advanced biomaterial technologies for tissue regeneration using 3D bioprinting, providing insights into futuristic healthcare technologies and regenerative medicines. The lecture series will conclude with Professor S. Dasappa, Advisor to Interdisciplinary Center for Energy Research and Professor at Center for Sustainable Technology, Indian Institute of Science, Bangalore, who will speak on renewable and green energy technologies for sustainable future, reinforcing the critical role of clean and renewable energy solution in achieving sustainable goals. I'm happy to note that there is a overwhelming response from various colleges as well as the university in and around Bangalore taking part in this particular lecture series, reflecting the wide academic outreach and relevance. I urge our students to actively participate in all the sessions, engage with the experts, ask questions, and make some more enriching learning opportunity. I wish the two-day lecture series of on science and technology for sustainable future a grand success. Thank you.
ಧನ್ಯವಾದಗಳು ಮೇಡಮ್. ಈ ಕಾರ್ಯಕ್ರಮವನ್ನು ವೇದಿಕೆಯ ಮೇಲೆ ಆಸೀನರಾಗಿರುವ ಎಲ್ಲ ಗಣ್ಯರು ಸಾಂಕೇತಿಕವಾಗಿ ಸಸಿಗೆ ನೀರೆರೆಯುವ ಮೂಲಕ ಉದ್ಘಾಟಿಸಬೇಕೆಂದು ಕೋರುತ್ತೇನೆ.
ಎಲ್ಲರಿಗೂ ಧನ್ಯವಾದಗಳು. ಈಗ ತಾನೇ ಕಾರ್ಯಕ್ರಮವನ್ನು ಉದ್ಘಾಟಿಸಿದ ಮಾನ್ಯ ವಿಧಾನ ಪರಿಷತ್ ಸದಸ್ಯರು, ಮಾಜಿ ವಿಜ್ಞಾನ ಮತ್ತು ತಂತ್ರಜ್ಞಾನ ಸಚಿವರು ಹಾಗೂ ಗೋಕುಲ ವಿದ್ಯಾಸಂಸ್ಥೆಯ ಅಧ್ಯಕ್ಷರು ಆದಂತಹ ಡಾಕ್ಟರ್ ಎಂ.ಆರ್. ಸೀತಾರಾಮ್ ಅವರು ಉದ್ಘಾಟನಾ ಭಾಷಣವನ್ನು ನೆರವೇರಿಸಿಕೊಡಬೇಕಾಗಿ ಕೋರುತ್ತೇನೆ.
ಎಲ್ರಿಗೂ ನಮಸ್ಕಾರ. ಗುಡ್ ಮಾರ್ನಿಂಗ್ ಟು ಆಲ್ ಆಫ್ ಯು. Chairman of this function, Dr. Raja Sahab, ex-Vice Chancellor of Tumkur University and the head of our postdoctoral programs in Ramaya Institute of Technology, Dr. Raja Sahab, Dr. Anand, the was devised so that this mobile planetarium could go into the schools, into the compound of the schools, because children will not know how a solar eclipse happens, lunar eclipse happens, the galaxy, because there is a lot of blind feelings that if there is a solar eclipse, you should not come out of the house. Lunar eclipse, you should not. Various other practices involved with that. So if the children come to know how or why a solar eclipse happens, or a lunar eclipse happens for that matter, when the Earth and Sun, Earth comes between Earth and Sun, or Moon comes between Sun and Earth, the eclipses happen. This is what the children had to be taught. So these mobile planetariums we installed and then sent them. I think as far as my knowledge goes, from 16 to around 2021, it continuously ran. After that, another government came and they stopped that. I think now we have started it, no? Again? Yeah, not yet. Started it. Again, after our government has come again, we have started it. This is only to increase the quest of happenings around us, whether the nature, the galaxy, the sea, water, and different environmental things given in nature. And I hope, these initiatives will slowly deviate the children from mundane and worthless things to focus themselves into research, development, and progress and prosperity automatically. So today it gives me a great pleasure that I've come here to inaugurate this 2-day lecture series co-sponsored by the Karnataka Science and Technology Academy and Ramaya College of Arts, Science and Commerce, and especially the Department of Biotechnology. I think how many colleges, other colleges, children have participated here? That is all Ramaya College. Other colleges. From which colleges you have come all? Yeah. Which one? JSS. From Mysore. Very good. You can stand and say no. Each one of you have come from outside. Okay. From outside Bangalore, Mysore has come. Anybody else? From North Karnataka, somewhere? I think we should also invite people, or you put it in Belgaum next, or Hubli. Okay. So these are all things where we try to get that keen interest in nature. And no department better than biotechnology, biomedical sciences, and these departments will take up this interest. And in future, I think, the bent on research, development, progress should be foremost in the minds of the children who are coming off. Let us not waste our time in, dividing society and deviating from mainstream of progress, being science-oriented, technology-oriented. And now you all know that artificial intelligence, machine learning, data sciences is a thing to come now. And unless you are exposed to these things, it becomes difficult for you to understand what is a fake, deep fake, or a real image. So even in the colleges where, be it, it is my opinion, of course, the academicians are all sitting here, I don't know what you say, but everybody should know what is artificial intelligence and how to use it to help humanity, not to harm humanity. Because these days you see cyber crimes have increased so much. Unless you know what cyber crime is, you'll be cheated in the course of time when you become big and start earning your money. People always give you these spam calls, warnings. Airtel gives you warnings when the phones come. This is a spam call. These things everybody should know. And unless you know, you will not be able to teach where the children should be exposed to all these things. And how many of you are speaking here today, today and tomorrow, participating? No, not the singing. I'm talking about, you're all listening only. You should also give chance to 2, 3 innovative children to speak also. Okay. After the end of this, is it? Okay, good. So, I wish all the participants all the best, and I think you should gain most from such lecture series, because it will open up your minds to see that what a lot of work we are there here to do. We can do a lot of things. We can even do creative things. Understanding nature itself, the man has not done fully till today. According to my perception, we are still understanding and learning what nature is. And we have only been trying to pollute the nature, pollute the atmosphere, rather than trying to understand what actual nature is, what geography is, what ecology is, and the environment is. So with these few words, I think that all of you should get the best out of this 2 days lecture series. And I think the initiative of the Science and Technology Department, I think, what is your outlay this year? You got this year? Crores. Okay, you are using it on this series only, like this only you can do it, no? No, forget the capital expenditure. That we will give special. Very less. How many of you are there on the rolls of 1-5? Okay. And we'll see that in this budget. I did about 150 crores I gave that time. Lot of money. We'll see that. Now we'll try to see that increases, but I was not happy the way you spend the money. That mobile planetarium was such a novel experiment which I gave. By now you could have given education to all children of all government schools in the entire state. Oh, yeah, I know, but you can also partner with them, no? Nothing stops you from partnering with them. Anyway, see, this is what I think, always I believe we should do something when we can do it. So, I did not ask anybody, I did not ask any experts when I was in Minister of Science and Technology, Planning and Statistics, because we get an opportunity. We try to help society so that you leave your mark, and that keeps on going, helping generations after generations. And I think all of you also in future, once you graduate and come out, some people will become postgraduates and enter research and things like that. After that, you also should focus on such things. With these few words, I think I've taken more time than what I was allotted. I think I was allotted about 5 or 10 minutes. So anyway, wishing all the children who are listening to the lecture series, and also the senior people who are addressing, and our Chandrayappa is also there. He was HOD of my biotechnology department in Ramaya Institute of Technology, and now he's in the College of Arts, Science and Commerce. He's a very senior man, but more on research, more on development. You can also, is he talking today? People like you also should talk after them. There's some time, do it. All the best to you, and I thank Raja Saab, the chairman of the Karnataka Science and Technology Academy, Dr. Anand, and all the principal, all the Department of Biotechnology in Ramaya College of Arts, Science and Commerce, and all of you for having invited me. Because after having granted, today I've come to see this auditorium, and I'm really happy that in a good place they have built it and it's being put to good use by everybody. Thank you. God bless you.
Thank you, sir, for that insightful address. We are truly honored to have you as the chief guest for our special lecture series. ಉದ್ಘಾಟನಾ ಭಾಷಣವನ್ನು ಮಾಡಿದ ಡಾಕ್ಟರ್ ಎಂಆರ್ ಸೀತಾರಾಮ್ ರವರನ್ನು ಸನ್ಮಾನಿಸಬೇಕಾಗಿ ಅಧ್ಯಕ್ಷರನ್ನು ಹಾಗೂ ಗೌರವಾನ್ವಿತ ಅತಿಥಿ ಗಣ್ಯರನ್ನು ಕೋರುತ್ತೇನೆ.
ಡಾಕ್ಟರ್ ಎಂ.ಆರ್. ಸೀತಾರಾಮ್ ಅವರು ಗೋಕುಲ ಎಜುಕೇಷನ್ ಫೌಂಡೇಶನ್ನ ಅಧ್ಯಕ್ಷರು ಹಾಗೂ ಕರ್ನಾಟಕ ಸರ್ಕಾರದ ವಿಧಾನ ಪರಿಷತ್ ಸದಸ್ಯರು, ಖ್ಯಾತ ಶಿಕ್ಷಣ ತಜ್ಞ ಹಾಗೂ ಸಮಾಜ ಸೇವಕ ಡಾಕ್ಟರ್ ಎಂ.ಎಸ್. ರಾಮಯ್ಯ ಅವರ ಪುತ್ರರಾದ ಅವರು ಶಿಕ್ಷಣ, ಸಾರ್ವಜನಿಕ ಸೇವೆ ಮತ್ತು ಸಮಾಜ ಕಲ್ಯಾಣ ಕ್ಷೇತ್ರಗಳಲ್ಲಿ ತಮ್ಮ ತಂದೆಯ ಪರಂಪರೆಯನ್ನು ಮುಂದುವರಿಸುತ್ತಿದ್ದಾರೆ. ಮಾಜಿ ಸಚಿವರಾಗಿರುವ ಡಾಕ್ಟರ್ ಸೀತಾರಾಮ್ ಅವರು ರಾಮಯ್ಯ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆಗಳ, ವಿಶೇಷವಾಗಿ ರಾಮಯ್ಯ ಇನ್ಸ್ಟಿಟ್ಯೂಟ್ ಆಫ್ ಟೆಕ್ನಾಲಜಿಯ ಅಭಿವೃದ್ಧಿಗೆ ಮಹತ್ವದ ಕೊಡುಗೆ ನೀಡಿದ್ದಾರೆ. ಅವರ ನಾಯಕತ್ವವು ಶೈಕ್ಷಣಿಕ ಶ್ರೇಷ್ಠತೆ ಮತ್ತು ಸಮಾಜ ಸೇವಾ ಅಭಿವೃದ್ಧಿಗೆ ಸಮರ್ಪಿತವಾಗಿದೆ.
ಎಲ್ಲರಿಗೂ ಧನ್ಯವಾದಗಳು. ವೇದಿಕೆಯ ಮೇಲೆ ಆಸೀನರಾಗಿರುವ ಗೌರವಾನ್ವಿತ ಅತಿಥಿಗಳಾದ ಡಾಕ್ಟರ್ ಪಾರ್ಶ್ವನಾಥ್ ಸಿಇ, ಗೋಕುಲ ಎಜುಕೇಷನ್ ಫೌಂಡೇಶನ್ ರವರಿಂದ ಗೌರವಾನ್ವಿತ ಅತಿಥಿಗಳ ಭಾಷಣ. All of you kindly stand. ವೇದಿಕೆಯ ಮೇಲೆ ಆಸೀನರಾಗಿರುವ ಗೌರವಾನ್ವಿತ ಅತಿಥಿಗಳಾದ ಡಾಕ್ಟರ್ ಪಾರ್ಶ್ವನಾಥ್ ಸಿಇ, ಗೋಕುಲ ಎಜುಕೇಷನ್ ಫೌಂಡೇಶನ್ ರವರಿಂದ ಗೌರವಾನ್ವಿತ ಅತಿಥಿಗಳ ಭಾಷಣ.
ಇವತ್ತಿನ ಈ ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಆಗಮಿಸಿರ್ತಕ್ಕಂಥ ತಮ್ಮೆಲ್ಲರಿಗೂ ವಂದಿಸಿ, ಮುಖ್ಯವಾಗಿ ಇವತ್ತಿನ honorable guests who are on the dais, and our minister has just now, our chairman has just now left after delivering his inaugural address. So at the outset, I should be thankful to all the guests and also all the speakers who will be participating in this program for the next today and tomorrow. So as was rightly pointed out by our honorable chairman, why this program is important, because today our children, not only from urban areas or from cities, even we should take it to our remote rural areas, so that our children will be aware of all the latest developments in science and technology. So with this in view, our department, our college, that is Ramaya College of Arts, Science and Commerce, today has organized this program jointly with the Science and Technology Academy. So in this program, they are going to highlight the various issues connected with science and technology for sustainable development. I think already you had got the first program in the morning for about an hour or so. So there I think our guest speaker has delivered it. And we have got some more speakers who are likely to address all of you today and tomorrow. At the outset, I think we are happy to know that this center has come up very well to see that all our children and various other organizations make use of this facility for development of science and technology in the state and in the country. So as you are aware, as already mentioned by our honorable chairman, that recent thing, that is artificial intelligence, the data science, and all that new subjects which has just come in. So that should be used in a befitting manner to enhance our knowledge and also the facility create out of that. I think today we are happy that apart from our Ramaya College, we have also few students coming from different parts of the state and also from different colleges. I think all the students should make use of this type of programs to enhance their knowledge and also the skills. So as already informed by our chairman, that is about 13 science centers are already functioning in the state of Karnataka. I had an opportunity of working with at least a few science centers. One in Raichur when I was the deputy commissioner there. Then second was also in Mangalore when I was there as CEO and also as the deputy commissioner, wherein we have an excellent facility provided. Pillukula, as already mentioned by our chairman, it has come up very well. The science center has come up very well in Mangalore, so also in Raichur. Like this, I think there are many other centers. That is what I should suggest, Raja Saab. You make use of all these centers to see that these type of programs are held in different parts of the state. So that will give an opportunity to people from all different parts of the state to make use of this facility. So with this, I think today we have this program being just inaugurated, and I am very happy to participate in this program and see that all of you can make best use of this. I think our honorable people who are coming from different subjects, different faculties, they are all going to give their lectures on various subjects. So make use of them and also actively participate to enhance your skills in science and technology so that it will be useful to the society at large. So with these few words, I once again wish to congratulate our Ramaya College of Arts, Science and Commerce, and Madam has already briefed about what are the activities for the next 2 days and who are all the speakers who will be participating. Similarly, all our colleagues, Dr. Karisitappa and Ramachandra, our finance officer, and Raja Saab, I think all of them are participating in this program in a big way. I think they will see that this program goes well for the next today and tomorrow, and all of you will be benefited from this type of interaction with various experts who will be addressing you. So thank you one and all. Jai Hind, Jai Karnataka. Namaskar.
ಧನ್ಯವಾದಗಳು ಸರ್. ಈಗ ಡಾಕ್ಟರ್
sincere advice to you all is try to understand the fundamentals. The tools can be picked up at any time, any point of time. Don't worry about the tools. The most of the studies you are concentrating on tools, learning languages, which is not so important. It is important, but it should be your second or third priority in your learning capabilities. So make yourself very strong in fundamentals. In the sense that unless you have the fundamental knowledge, the basic skill set, you will not be able to, conceive a problem which is faced by the society and its thinking behind and giving a wonderful solution which is in favor of societal welfare. So that is the first need wherein you have to learn you must be very thorough with fundamentals, principles of science and technology, and later on you can pick up tools. Now some of the challenges, the most of the sustainable problems we are facing, our society are facing, it is in terms of health, it is in terms of energy, and it is in terms of environment. So these are the things which are connected to each other. Your health is connected to environment, environment is connected to energy, and these three are the very basic approaches you should cultivate to adopt science and technology and innovations to come out with a wonderful solution to the society. Now, all these things are connected with depletion of natural resources, some of the challenges of whatever man-made problems, and augmenting those problems and coming out with wellness, or coming out with solutions which are good for the mankind is your problem. It is the challenge. Now, some of the technologies you have to adopt yourself. One of the important technology what we are talking about when you want to have problem solved or higher level thinking is 3D printing. You can create real models and study. Earlier, some experimental setup or creation of some model was a very big task, and you can save so much of time and come out with very good experimental setup or studies if you use 3D principle, 3D modeling. Then other technology is AI. It is a tool, as I said, you can learn or pick up at any time. But how can you use this? For example, artificial intelligence in managing power sector. We talk about intelligent grids, where we can minimize energy loss, we can improve the efficiency, and you can use a newer technology, newer innovations like AI tools, etc., and come out with solution. Now, to manage energy, you can automate everything. You can control based on the real data, based on the field data. You will be able to come out with a very efficient system in terms of minimizing the energy loss, in terms of scheduling the distribution, in terms of taking up the repair or maintenance work based on the real data, etc. So that is the technology, the power of AI you can use in the energy management. The other very challenging one, many of the countries are working in a large scale, is about carbon characterization or control and the storage. CCR they call it as. Okay, so the carbon emission from factories or industries can be stored in underground and minimize the effect of carbon on the environment. So that you can think about innovations. You can think about how to do this. The other challenging, interesting area is in terms of solar energy. Some of the energy efficient buildings have self-sustainable buildings we are coming out with. So one of the research area people we are working are solar windows. You coat solar windows with a special membrane. They act as energy cells. They can generate solar energy and use it for rebuilding. It is self-sustaining energy system. So you can think about that technology. The other area where you can contribute is about purification of water. We don't have a very efficient water or a sea water, you can get a pure water. Normally we use reverse osmosis or some nanotechnology. Now you think about a different technology using the tools like AI or big data analytics. How can you purify the water with minimum cost? And it could be affordable one, because there is a depletion in natural resources available. So we use extensively this. And you live in a society wherein very highly intensive society. You consume so much of energy and those kind of things. In urban area, there is another approach people are working on, is about vertical irrigation. In urban area, you can use this hydroponic and LED lights, grow vegetables, some of the organic leaves or those kind of things. So you can use the open space in urban area, so that there will be a less pressure on the irrigation on the land, and less pollution. Consumption water is minimum, so with minimum water content you can use efficiently the hydroponic irrigation or vertical irrigation. So these are some of the technologies you all should think about and come out with solution. These kind of approaches will make you very strong in your core area, and also you are solving the problems of common man. That's what sir was mentioning about. Any technology, anything you innovate should be to the welfare of the society, not for destruction. So these are some of the challenges you have because of ever-growing population across the globe, and the globe cannot change its dimensions. We cannot have more, earth or useful land. It cannot grow. So we should optimize the utilization, come out with technologies which are environmental friendly, which are good to human, and which are less harming, and with more of useful and long lasting. So any solution you work, any solution you think about in terms of the newer technology or energy, it has to be in those directions, in the direction of well-being or human welfare. So you think about these kind of technologies and science and try to grow that. Don't waste more on the services or the learning tools, etc. You must be very, very efficient in terms of understanding numerically powerful computational facilities, high funda things in physics or mathematics or biology or biotechnology, whatever it is. So they make you very strong inside in core. And then you can pick up all these tools, the machine learning, AI, whatever technology you come across. Another important area we are all trying to work in Ramaya is about semiconductors, AI applications in innovations, and also in computational efficiency in terms of quantum computing. Very large scale problems can be solved within no time, few seconds. So we are trying to open those kind of computational facility in Ramaya itself, so that it will be open to all the institutions. There could be some economic model for educational institution. So these are certain things you have to be very, very serious, and you have to spend the available tools and technology to your advantage. Many things, as sir was mentioning, honorable chairman, so many a times we try to misuse the technology. We try to misuse it for destructive things, which is not so. You have to use this technology for your own advantage and for future, as rightly put in your today's lecture series, sustainable future. So these are some of the core areas you can all work on this. And these disciplines, once you are fundamentally strong, you need not worry about the background from which you are coming. You are coming from physics or biology, or from engineering, or from only pure science. No. When you specialize yourself, you converge. You all become one, like your 12th standard. You are all together, right? Maybe 10th standard, not 12th standard, 10th standard. In 12th you have commerce, science, and all those. And you started diverting from there. So when you super specialize, you all converge. So you can contribute more. So that is the kind of future sitting in this hall. So I wish you all a very wonderful future. Thank you very much.
ಧನ್ಯವಾದಗಳು ಸರ್. ಈಗ ಶ್ರೀರಾಮಚಂದ್ರ, ಚೀಫ್ ಆಫ್ ಫೈನಾನ್ಸ್, ಗೋಕುಲ ಎಜುಕೇಷನ್ ಫೌಂಡೇಶನ್ ಅವರಿಂದ ಗೌರವಾನ್ವಿತ ಅತಿಥಿಗಳ ಭಾಷಣ.
Good morning, everybody. Respected dignitaries on the dais, Dr. Sitaram Sarji, Dr. Pashnath, Dr. Kstacademy, Dr. Rajashar, Dr. Pushpa Madam, and all the dignitaries present here, Dr. Anand, and also dear students. I'm happy to be part of this event. I'm not an academician. I can't give a good lecture. However, I would like to tell you that you are all going to be future scientists and researchers of this country. You have a bright future. Our economy is growing. There is a huge opportunity for all of you. Only my suggestion is, whenever you do research, whenever you do innovate new things, ensure that it is financially sustainable also. Unless you keep this in your mind, it may not give results. Many of our research activities and many of innovations don't come out of the lab because it is not financially sustainable. I think you should keep this also in mind as you grow in your career. All your research and innovation should have a financial aspect. It should ensure that it not even immediately, in the long term, it has to be financially sustainable. I only hope that all you keep this in mind. I request you to ensure that you take maximum benefit out of this 2 days lecture series. I wish all success for this series. Thanks for me calling me here. Thank you.
ಧನ್ಯವಾದಗಳು ಸರ್. ಗೌರವಾನ್ವಿತ ಅತಿಥಿಗಳಾದ ಡಾಕ್ಟರ್ ಪಾರ್ಶ್ವನಾಥ್, ಸಿಇ, ಗೋಕುಲ ಎಜುಕೇಷನ್ ಫೌಂಡೇಶನ್ ರವರನ್ನು ಸನ್ಮಾನಿಸಬೇಕಾಗಿ ಅಧ್ಯಕ್ಷರನ್ನು ಮತ್ತು ಅತಿಥಿ ಗಣ್ಯರನ್ನು ಕೋರುತ್ತೇನೆ. ಧನ್ಯವಾದಗಳು. ಈಗ ಗೌರವಾನ್ವಿತ ಅತಿಥಿಗಳಾದ ಡಾಕ್ಟರ್ ಕರಿಸಿದ್ದಪ್ಪ, ಚೀಫ್ ಅಕಾಡೆಮಿಕ್ ಅಡ್ವೈಸರ್, ಗೋಕುಲ ಎಜುಕೇಷನ್ ಫೌಂಡೇಶನ್ ರವರನ್ನು ಸನ್ಮಾನಿಸಬೇಕಾಗಿ ಅಧ್ಯಕ್ಷರನ್ನು ಮತ್ತು ಅತಿಥಿ ಗಣ್ಯರನ್ನು ಕೋರುತ್ತೇನೆ. ಧನ್ಯವಾದಗಳು. ಈಗ ಗೌರವಾನ್ವಿತ ಅತಿಥಿಗಳಾದ ಶ್ರೀ ರಾಮಚಂದ್ರ ಜಿ, ಚೀಫ್ ಆಫ್ ಫೈನಾನ್ಸ್, ಗೋಕುಲ ಎಜುಕೇಷನ್ ಫೌಂಡೇಶನ್ ರವರನ್ನು ಸನ್ಮಾನಿಸಬೇಕಾಗಿ ಅಧ್ಯಕ್ಷರನ್ನು ಮತ್ತು ಅತಿಥಿ ಗಣ್ಯರನ್ನು ಕೋರುತ್ತೇನೆ. ಧನ್ಯವಾದಗಳು. ಉಪನ್ಯಾಸ ಮಾಲಿಕೆ ಅಧ್ಯಕ್ಷತೆ ವಹಿಸಿರುವ ಡಾಕ್ಟರ್ ರಾಜಾಸಾಬ್ ಎ ಹೆಚ್ ಅಧ್ಯಕ್ಷರು KSTA ಇವರು ಅಧ್ಯಕ್ಷೀಯ ಭಾಷಣವನ್ನು ನೆರವೇರಿಸಿಕೊಡಬೇಕಾಗಿ ಕೋರುತ್ತೇನೆ.
ಎಲ್ಲರಿಗೂ ಮತ್ತೊಮ್ಮೆ ನಮಸ್ಕಾರಗಳು. ಟುಡೇ ಈಸ್ ಎ ವೆರಿ ಡೇ ಫಾರ್ ರಿಮೆಂಬರಿಂಗ್ in the days to come, because former minister and present MLA and also chairman of Gokula Education Society, Ramaya Group of Institutions, he came and inaugurated the very good themed conference, lecture series. And I would like to say here that in addition to our chairman, other 3 persons, guests of honor, Dr. Kstacademy, Dr. Parshanath and Shri Ramachandra ji came here. In my view, these 3 for Gokula Education Foundation, they are like Trimurtis. ಇವರು ತ್ರಿಮೂರ್ತಿಗಳು. ಪೂರ್ತಿ ಸೊಸೈಟಿಯನ್ನು, ಎಜುಕೇಷನ್ ಫೌಂಡೇಶನ್ನ ನಡೆಸೋದು ಇವರೇ. They are so busy people actually. So many people wait for them every day to get their files approved, etc., etc. But today, all 3 of them have come. It is probably because they have great love towards, affection towards the academy, as well as the function organized by Ramaya College of Arts, Science and Commerce. And there is a overwhelming response. More than 170 registrations, you may not believe. Our hall, this capacity is about 120 students, and the remaining students they are sitting in our video room because it is there also the program is live coming. So others are there. So more than 150, 160 students, they are seeing this program. In addition to this, all over Karnataka and other people, they also, that facility is there. It is broadcasted there also. And we have YouTube channel also. In the YouTube also these things will come. So it is a good program. Kstacademy, I was not able to introduce these 3 persons in my first speech because our chairman was to go early. I introduced chairman, but intentionally, although I welcomed them, I didn't introduce them. It is my responsibility to introduce them. Dr. Kstacademy sir, I know since a decade or so. He was Vice Chancellor of VTU. During his tenure, the VTU got very good name. And really, I am telling, because earlier to that, there were so many problems in VTU, Vishweshwarya Technological University. When Sir Dr. Kstacademy took over the charge, he made the phenomenal changes. He brought in phenomenal changes. Everything was streamlined. And he is known for his very good administration. And one of my close friend who was controller of exam, registrar, evaluation, worked under him. So in order to understand how the Vice Chancellor is, we should ask the students and the officers. They will give the correct picture. That my friend was telling, sir, ಅವರು ತುಂಬ ಡೌನ್ ಟು ಅರ್ತ್ ಕಂಟ್ರಿ, ಸರಿ ಇದ್ದಿದ್ದನ್ನು ಸರಿಯಾಗಿ ಮಾಡೇ ಮಾಡ್ತಾರೆ, ಯಾರು ಹೇಳಿದ್ರು ಚೇಂಜ್ ಆಗೋದಿಲ್ಲ. ಇವನ್ ಸರ್ಕಾರದಿಂದ ಒತ್ತಡ ಬಂದರೂ ಅವ್ರ ಮನಸ್ಸು ಚೇಂಜ್ ಮಾಡೋದಿಲ್ಲ, ಸರಿ ಇದ್ದಿದ್ದ ಸರಿ ಅಂಥ ಒಂದು ಒಳ್ಳೆ ಪರ್ಸನಾಲಿಟಿ ಅಂತ. ದಿಸ್ ಇಸ್ ವಾಟ್ ದಿ ಫೀಡ್ಬ್ಯಾಕ್ ಐ ಗಾಟ್ ಫ್ರಮ್ ಹಿಮ್. ಆ್ಯಂಡ್ ಫ್ರಮ್ ದಟ್ ಡೇ ಆನ್ವರ್ಡ್ಸ್ ಐ ಡೆವಲಪ್ಡ್ ಅ ಗುಡ್ ರ್ಯಾಪೋರ್ಟ್ ವಿತ್ ಹಿಮ್. And we are good friends. We discuss, we meet almost now and then. We discuss academic matters, administrative matters, and he is really an asset to the Ramaya group of institutions. The second one is Dr. Parshanath. I know Dr. Parshanath last 40 years, IAS officer. 40 years, can you believe? Because we were classmates in MSc, Mysore University, and we were also research mates. We worked in the same laboratory. So ever since our student days, we know one another. He knows my way, philosophy of life. I know his way of thinking, his good nature. I have always thought myself when I am alone, how to imbibe the good behavioral traits of Dr. Parshanath. It is so difficult. I behave like an ordinary man. He is above ordinary person, very noble person. I have never seen him becoming angry on anybody. I have 40 years. I have never seen him becoming angry on anybody or talking ill about others either in their presence or absence. This is the character which I observed in him. Very noble person. It is good to emulate him, but it is difficult to emulate him. This is my experience. Very wonderful person, highly cultured. He comes from a highly cultured family. And he passed when he was doing PhD, he passed the KS. And then became officer, KS officer. Worked as a DC, CEO, Jilla Panchayat DC in Mangalore as well as in Raichur. He made a good name. And later he became secretary of the KPSC. And then he was the member of the KPSC also. Then afterwards, he took his retirement, and Ramaya Gokula Education, the same Sitaram sir and Jairam sir, they called him to work as CEO, Chief Executive Officer. He is the Chief Executive Officer of all the institutions coming under Ramaya Group of institutions. That is about Dr. Parshanath. What I like in him is his philosophy of life. And Ramchandra sir, always Dr. Ramchandra sir, Shri Ramchandra ಅವರು ಡಾಕ್ಟರ್ ಪಾರ್ಶ್ವನಾಥ್, ಡಾಕ್ಟರ್ ಕರಿಸಿದ್ದಪ್ಪ ಯಾವಾಗಲೂ ಜೊತೆಗಿರ್ತಾರೆ. ಇಷ್ಟು ಸ್ಮೂತ್ ಅಡ್ಮಿನಿಸ್ಟ್ರೇಷನ್ ಕೊಡ್ತಿದ್ದಾರೆ. ಈ ಈಸ್ ಚೀಫ್ ಆಫ್ ಫೈನಾನ್ಸ್. ಯಾವುದು ಗೋಕುಲ ಎಜುಕೇಷನ್ ಚೀಫ್ ಆಫ್ ಫೈನಾನ್ಸ್. ಅರ್ಲಿಯರ್ ಆಲ್ಸೋ he worked as finance in the finance department, financial advisor, auditing, etc. He has well known, very deep knowledge in his field of specialization. And these three people are running the institute, Gokula Education Foundation, aspects of Gokula Education Foundation in a very excellent way and in a befitting manner. And Dr. Pushpa Madam, your beloved principal, I have seen her as principal. Even before she became principal also we have seen her. She is the same. A person with inclusive qualities. After she became principal, I am of the opinion that all her colleagues, the students, felt very happy because she has that inclusive philosophy. She always talks to everybody, takes them into confidence. People start working themselves. That is how Ramaya College of Arts, Science and Commerce, they became institutional members. They are that institution, Ramaya College of Arts, Science, Commerce, is an institutional member of KSTA. The institutional members have special privileges. We invite them for all the conferences. We give them some financial assistance to organize the conferences either here or in their place. And if they organize conferences here, for example, today's example, all the facilities here, they are at their disposal. 2 days. They can use all the facilities here. This is the privilege. Madam is doing extremely well as principal of that college, and it is an autonomous college. Now we are planning, I am telling Madam, even for PhD program they should become autonomous. They should write to Bangalore University, get the autonomous position for PhD program from Bangalore Central University. Then we can start the full-time PhD program in a more befitting and useful way. This is what I am suggesting to them. The KSTA is always with them, and we welcome the students of Ramaya as well as the staff of Ramaya. They have worked yesterday also, they were here till late evening to arrange this program, and they did very well. I didn't expect that program will go so well today, but yesterday I was telling them, do this, do this, do this, do this. They did it. So I should thank the faculty and other staff members of Ramaya College of Science, Arts and Commerce. Similarly, our people also, KSTA staff, I always appreciate them. They always work in a team spirit. Yesterday night they have not gone their homes. I went, but they did all the work yesterday here. They stayed here. So this is the way the KSTA works. And now you are the students. I request you to become the associate members of our KSTA. If you become associate members, you can come here anytime. You can participate in the program. You will get our Vijnana Loka Torsi. Vijnana Loka, free of cost you will get 6 issues in a year. This Vijnana Loka is the bunch, contains bunch of the science articles, especially in Kannada. Very valuable articles are there. Some samples we have kept. If you want, you can take them. And you can become associate member. You will get Vijnan Loka. And if you are good in communicating science in Kannada, you are most welcome. You can contribute the articles to Vijnan Loka. It's a very standard article in Kannada. It will go to all schools in Karnataka, believe, all colleges in Karnataka. So PU colleges also, it is sent. So if you write an article, people will come to know about your capabilities. So with these few words, I stop here. This is a good lecture series. Yesterday I met one agriculture professor, and especially he drew my attention to one point about artificial intelligence. He said he has Arakanat garden in Shumaga district near Davangere. Rich guy. He showed me photographs, and he has made a PPT presentation also in Goa, ICR conference day before yesterday. In that he showed me there is a firm in Pune. If you send the coconut garden or Arakanat garden or any field survey number to that industry startup, they will survey your field through satellite and find out the soil conditions of your garden. They will find out what is the content of nitrogen, phosphorus, calcium, etc., minor and major nutrients through satellite by way of AI technology, and they will tell you have to apply the next fertilizer regime. The proportions they will give. In these proportions you have to apply. They will give recommendation based on the soil characters. So instead of testing the soil in laboratory, AI will test. And they have compared also physical testing, laboratory testing and AI data. They match with one another. So it is not wrong what data it is giving. And he has grown Arakanut in about 3 acres. And last year he applied the 8 recommendations and fertilizers. Before application of this technology, in the cultivation, per acre he was getting a net profit of 3 lakhs. Okay. Now after the AI recommendation and fertilizer application, he is getting 10 lakhs per acre. So 3 to 10 lakh is almost 3-fold increase. And he said, these scientists will sit in the laboratory and do so much of work. It is useless, he said. Now technology has come. One should use the technology for the welfare of the human beings. It may be agriculture, it may be medicine, or it may be some other technology. They should use it, they should apply it. And ordinary Arakanut farmer is benefited by AI. This is how knowledge should reach the farmers. And last month we did a big conference here on quantum technology. Like this hall was full. So maybe in next 5 years, quantum and AI are going to help the humankind to a greater extent. In all fields, quantum will come. In all fields, even medical diagnostics also, quantum will come. In space technology also, quantum will come. AI and quantum merge, and then if we use judiciously the AI and quantum technology, it is possible to apply that knowledge to the welfare of the human beings. So with these few words, I stop here. Thank you very much for your patience.
ಧನ್ಯವಾದಗಳು ಸರ್. ಈ ಅಭೂತಪೂರ್ವ ಉಪನ್ಯಾಸ ಮಾಲಿಕೆಯಲ್ಲಿ ಪಾಲ್ಗೊಂಡವರೆಲ್ಲರಿಗೂ ವಂದನೆಗಳನ್ನು ಸಲ್ಲಿಸಲಿದ್ದಾರೆ ಅಕಾಡೆಮಿಯ ಮುಖ್ಯ ಕಾರ್ಯನಿರ್ವಾಹಕ ಅಧಿಕಾರಿಗಳಾದ ಡಾಕ್ಟರ್ ಆನಂದ್ರವರು.
ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. ಎ ವೆರಿ ಗುಡ್ ಮಾರ್ನಿಂಗ್ ಟು ಆಲ್. ಇಂದು ನಮ್ಮ ಕೋರಿಕೆಯನ್ನು ಮನ್ನಿಸಿ ಈ ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ಭಾಗವಹಿಸಿ ಈ ಕಾರ್ಯಕ್ರಮದ ಮೆರುಗನ್ನು ಹೆಚ್ಚಿಸಿ ಉದ್ಘಾಟನೆಯನ್ನು ನೆರವೇರಿಸಿಕೊಟ್ಟ ಗೋಕುಲ ಶಿಕ್ಷಣ ಪ್ರತಿಷ್ಠಾನದ ಗೌರವಾನ್ವಿತ ಅಧ್ಯಕ್ಷರು, ವಿಧಾನ ಪರಿಷತ್ ಸದಸ್ಯರು ಹಾಗೂ ಯೋಜನೆ ಸಾಂಖ್ಯಿಕ ಹಾಗೂ ವಿಜ್ಞಾನ ಮತ್ತು ತಂತ್ರಜ್ಞಾನ ಮಾಜಿ ಸಚಿವರು ಆದ ಡಾಕ್ಟರ್ ಎಂ.ಆರ್. ಸೀತಾರಾಮ್ ಸರ್ ಅವರಿಗೆ ಅವರ ಅನುಪಸ್ಥಿತಿಯಲ್ಲಿ ಅಕಾಡೆಮಿಯ ಪರವಾಗಿ ಎಂ.ಎಸ್. ರಾಮಯ್ಯ ಕಾಲೇಜ್ ಪರವಾಗಿ ಹಾಗೂ ನಿಮ್ಮೆಲ್ಲರ ಪರವಾಗಿ ಹೃತ್ಪೂರ್ವಕ ಧನ್ಯವಾದಗಳನ್ನು ಅರ್ಪಿಸುತ್ತೇನೆ. ಹಾಗೆಯೇ ಗೋಕುಲ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆಯ ನಿರ್ದೇಶಕರು, ಟ್ರಸ್ಟಿಗಳು ಅವರಿಗೆ ಅವರ ಸಹಕಾರಕ್ಕೆ ಧನ್ಯವಾದಗಳನ್ನು ಅರ್ಪಿಸುತ್ತೇನೆ. ಈ ಸಮಾರಂಭದ ಅಧ್ಯಕ್ಷತೆಯನ್ನು ವಹಿಸಿಕೊಂಡು ಸಮಾರಂಭದ ಯಶಸ್ಸಿಗೆ ಕಾರಣರಾದ ಅಕಾಡೆಮಿಯ ಮಾನ್ಯ ಅಧ್ಯಕ್ಷರಾದ ಪ್ರೊಫೆಸರ್ ರಾಜಾಸಾಬ್ ಎಚ್ ಇವರಿಗೆ ತಮ್ಮೆಲ್ಲರ ಪರವಾಗಿ, ರಾಮಯ್ಯ ಕಾಲೇಜ್ ಪರವಾಗಿ ಹಾಗೂ ವೈಯಕ್ತಿಕವಾಗಿ ಧನ್ಯವಾದಗಳನ್ನು ಅರ್ಪಿಸುತ್ತೇನೆ. ಈ ಸಮಾರಂಭದಲ್ಲಿ ಗೌರವಾನ್ವಿತ ಅತಿಥಿಗಳಾಗಿ ಭಾಗವಹಿಸಿದ್ದ KPSCನ ಮಾಜಿ ಸದಸ್ಯರು ಹಾಗೂ ಗೋಕುಲ ಶಿಕ್ಷಣ ಪ್ರತಿಷ್ಠಾನದ ಮಾನ್ಯ ಮುಖ್ಯ ಕಾರ್ಯನಿರ್ವಾಹಕರಾದ ಡಾಕ್ಟರ್ ಪಾರ್ಶ್ವನಾಥ್ HV IAS ಇವರಿಗೆ ಕರ್ನಾಟಕ ವಿಜ್ಞಾನ ಮತ್ತು ತಂತ್ರಜ್ಞಾನ ಅಕಾಡೆಮಿ, ರಾಮಯ್ಯ ಕಾಲೇಜು ಹಾಗೂ ತಮ್ಮೆಲ್ಲರ ಪರವಾಗಿ ಹೃತ್ಪೂರ್ವಕ ಧನ್ಯವಾದಗಳನ್ನು ಅರ್ಪಿಸುತ್ತೇನೆ. ಹಾಗೆ ಈ ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ಗೌರವಾನ್ವಿತ ಅತಿಥಿಗಳಾಗಿ ಭಾಗವಹಿಸಿದ್ದ ಗೋಕುಲ ಶಿಕ್ಷಣ ಪ್ರತಿಷ್ಠಾನದ ಮುಖ್ಯ ಶೈಕ್ಷಣಿಕ ಸಲಹೆಗಾರರು ಹಾಗೂ ವಿಶ್ವೇಶ್ವರಯ್ಯ ತಾಂತ್ರಿಕ ವಿಶ್ವವಿದ್ಯಾಲಯದ ವಿಶ್ರಾಂತ ಕುಲಪತಿಗಳಾದ ಡಾಕ್ಟರ್ ಕರಿಸಿದ್ದಪ್ಪರವರಿಗೆ ಅಕಾಡೆಮಿ ಪರವಾಗಿ, ರಾಮಯ್ಯ ಕಾಲೇಜ್ ಹಾಗೂ ನಿಮ್ಮೆಲ್ಲರ ಪರವಾಗಿ ಧನ್ಯವಾದಗಳನ್ನು ಅರ್ಪಿಸುತ್ತೇನೆ. ಈ ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ಭಾಗವಹಿಸಿರುವ ಗೋಕುಲ ಶಿಕ್ಷಣ ಪ್ರತಿಷ್ಠಾನದ ಚೀಫ್ ಆಫ್ ಫೈನಾನ್ಸ್ ಆದ ಶ್ರೀ G. ರಾಮಚಂದ್ರ ಅವರಿಗೆ ಅಕಾಡೆಮಿಯ ಪರವಾಗಿ ನಿಮ್ಮೆಲ್ಲರ ಪರವಾಗಿ ಧನ್ಯವಾದಗಳನ್ನು ಅರ್ಪಿಸುತ್ತೇನೆ. ಈ 2 ದಿನಗಳ ಉಪನ್ಯಾಸ ಮಾಲೆಯ ಪ್ರಮುಖ ರೂವಾರಿಯಾದ MS ರಾಮಯ್ಯ ಕಲಾ ವಿಜ್ಞಾನ ಹಾಗೂ ವಾಣಿಜ್ಯ ಕಾಲೇಜಿನ ಪ್ರಾಂಶುಪಾಲರಾದ ಡಾಕ್ಟರ್ ಪುಷ್ಪ ಇವರಿಗೆ ಅಕಾಡೆಮಿಯ ಪರವಾಗಿ ನಿಮ್ಮೆಲ್ಲರ ಪರವಾಗಿ ಧನ್ಯವಾದಗಳನ್ನು ಅರ್ಪಿಸುತ್ತೇನೆ. ಈ 2 ದಿನ ಕಾರ್ಯಾಗಾರದ ಉಪನ್ಯಾಸ ಮಾಲೆಯ ಕನ್ವೀನರ್ಗಳಾದ ಡಾಕ್ಟರ್ ಲಕ್ಷ್ಮೀಕಾಂತ್ RN, ಡಾಕ್ಟರ್ ಪ್ರಸನ್ನ ಕುಮಾರ್ ಇವರಿಗೆ ಧನ್ಯವಾದಗಳನ್ನು ಅರ್ಪಿಸುತ್ತೇನೆ. ಹಾಗೆ ಈ ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ಭಾಗವಹಿಸಿರುವ ರಾಮಯ್ಯ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆಯ ರಿಸರ್ಚ್ ಹೆಡ್ ಆದ ಪ್ರೊಫೆಸರ್ ಚೆನ್ನರಾಯಪ್ಪರವರಿಗೆ ಅಕಾಡೆಮಿಯ ಪರವಾಗಿ ನಿಮ್ಮೆಲ್ಲರ ಪರವಾಗಿ ಧನ್ಯವಾದಗಳನ್ನು ಅರ್ಪಿಸುತ್ತೇನೆ. ಈ ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ಭಾಗವಹಿಸಿದ್ದ ಅಕಾಡೆಮಿಯ ಮಾನ್ಯ ಸದಸ್ಯರುಗಳಾದ ಡಾಕ್ಟರ್ ಪ್ರಭಾಕರ್ ಸಂಗೂರ್ಮಠ್, ನಿವೃತ್ತ ನಿರ್ದೇಶಕರು, ಹಟ್ಟಿ ಗೋಲ್ಡ್ ಮೈನ್ಸ್, ಹಾಗೂ ಡಾಕ್ಟರ್ ಚಂದ್ರಶೇಖರ್ ಬಿರ್ದರ್ ಸರ್ ಅವರಿಗೆ ಅಕಾಡೆಮಿಯ ಪರವಾಗಿ ನಿಮ್ಮೆಲ್ಲರ ಪರವಾಗಿ ಧನ್ಯವಾದಗಳನ್ನು ಅರ್ಪಿಸುತ್ತೇನೆ. ಈ ಉಪನ್ಯಾಸ ಮಾಲೆ ಹಾಗೂ ಈ ಸಮಾರಂಭದ ಯಶಸ್ಸಿಗೆ ಕಾರಣರಾದ ರಾಮಯ್ಯ ಕಾಲೇಜಿನ ವಿಜ್ಞಾನ ವಿಭಾಗದ ಮುಖ್ಯಸ್ಥರು, ಅಧ್ಯಾಪಕರು ಮತ್ತು ಇತರೆ ಬೋಧಕೇತರ ಸಿಬ್ಬಂದಿಗಳಿಗೆ ಧನ್ಯವಾದಗಳನ್ನು ಅರ್ಪಿಸುತ್ತೇನೆ. ಈ ಕಾರ್ಯಾಗಾರದ ಛಾಯಾಚಿತ್ರಣ ಹಾಗೂ ಛಾಯಾಗ್ರಹಣ ಚಿತ್ರಣವನ್ನು ನಡೆಸುತ್ತಿರುವ ರಾಮಯ್ಯ ಕಾಲೇಜಿನ ಮೀಡಿಯಾ ಟೀಮ್ಗೆ ಧನ್ಯವಾದಗಳನ್ನು ಅರ್ಪಿಸುತ್ತೇನೆ. ಈ ಕಾರ್ಯಕ್ರಮದ ಯಶಸ್ಸಿಗೆ ಕಾರಣರಾದ ಅಕಾಡೆಮಿಯ ಎಲ್ಲ ಸಹೋದ್ಯೋಗಿಗಳು, ರಾಮಯ್ಯ ಕಾಲೇಜಿನ ಆಡಳಿತ ಹಾಗೂ ಹಣಕಾಸು ವಿಭಾಗ, ಟ್ರಾನ್ಸ್ಪೋರ್ಟ್ ವಿಭಾಗ ಇವರಿಗೆ ಧನ್ಯವಾದಗಳನ್ನು ಅರ್ಪಿಸುತ್ತೇನೆ. ಈ ಉಪನ್ಯಾಸ ಮಾಲೆ ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ಪ್ರತಿನಿಧಿಗಳನ್ನು ನಿಯೋಜಿಸಿರುವ ಅಕಾಡೆಮಿಯ ಸಾಂಸ್ಥಿಕ ಸದಸ್ಯತ್ವ ಹೊಂದಿರುವ ಕಾಲೇಜುಗಳು ನೃಪತುಂಗ ವಿಶ್ವವಿದ್ಯಾನಿಲಯ, ರೇವಾ ವಿಶ್ವವಿದ್ಯಾನಿಲಯ, ಕ್ರಿಸ್ತು ಜಯಂತಿ ಯೂನಿವರ್ಸಿಟಿ, ಆಕ್ಸ್ಫರ್ಡ್ ಯೂನಿವರ್ಸಿಟಿ, JSS ಹೈಯರ್ ಎಜುಕೇಷನ್ ರಿಸರ್ಚ್ ಮೈಸೂರು, ICAR, NBAIR ಇಲ್ಲಿಂದ ಬಂದಿರೋ ಪ್ರತಿನಿಧಿಗಳಿಗೆ ಧನ್ಯವಾದಗಳು. ಈ ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ಭಾಗವಹಿಸಿರುವ ಎಲ್ಲ ಪ್ರತಿನಿಧಿಗಳಿಗೆ ಧನ್ಯವಾದಗಳು. ಈ ಕಾರ್ಯಕ್ರಮವನ್ನು ನಿರೂಪಣೆಯನ್ನು ನಡೆಸಿಕೊಟ್ಟಿರೋರಿಗೆ ಧನ್ಯವಾದಗಳು. ಇಲ್ಲಿ ನೆರೆದಿರುವ ಎಲ್ಲ ಗಣ್ಯರಿಗೆ, ಆಹ್ವಾನಿತರಿಗೆ ಹಾಗೂ ಎಲ್ಲ ಸಭಿಕರಿಗೆ ಧನ್ಯವಾದಗಳು.
ಧನ್ಯವಾದಗಳು ಸರ್. ಇದರೊಂದಿಗೆ ಈ ಉದ್ಘಾಟನಾ ಸಮಾರಂಭ ಮುಕ್ತಾಯವಾಗುತ್ತದೆ. ಎಲ್ಲರಿಗೂ ಧನ್ಯವಾದಗಳು. I request all the students to be seated as we will begin with the technical session 2.
Good afternoon, everyone. A very warm welcome to one and all present here. We now move on to the second session of the lecture series. I request Professor Chandrappa sir to welcome our distinguished speaker, Dr. Professor Chandrasekhar M. Biradar, with a sapling. Thank you, sir. It gives me immense pleasure to introduce our speaker for the second session, Professor Chandrasekhar M. Biradar. Professor Dr. Chandrasekhar M. Biradar is a globally acclaimed Earth system scientist, landscape ecologist, and green growth architect with 30 plus years of work across 50 plus countries. His contributions span digital augmentation, regenerative agriculture, multifunctional agroforestry, springshed restoration, biodiversity conservation, natural farming, and large-scale agri-food system transformation. After decades abroad, he returned to India to build the nation as the world's next soft power in the nature-based green economic growth. He is the architect of green economic growth and serves as a chairman and managing director of Global Green Growth Community of Practices, advancing India's leadership in nature-centric, science-led solutions. He has held senior roles across CGIAR centers, UN FAO, Rainforest Alliance, and global institutions. With 550 plus publications, he has designed transformative frameworks such as Nandi Krishi, Green Gram Swarajya, Green Growth in Drylands, Circular Village, Living Lands, Farm as Resort, Net Zero Living, Biodiversity, and Karma Credits. He currently chairs the global initiative on tree-based future foods and multifunctional landscapes and green economic growth, shaping pathways to sustainable development, land degradation neutrality, climate resilience, and a net zero nation. I now invite Professor to kindly take over this session and enlighten us with his knowledge. Over to you.
So good morning. Thank you very much. So thank you once again, and Chairman and all the dignitaries here. As this morning, Chief Guest Sitaram sir, they're very interesting. We are progressing very well in the science, technology, innovations, and AI. How that really became a sustainable, how a really sustainable world we are living. So let's go through the series of the innovations, and I focused on the new generations, really how this augmented science, technology, innovations, as well as the citizen science, they're going to drive the future of the India and the world. So context is very clear. Degradation accelerating in a very massive way. Number of reasons, we all aware of that, including the food we eat, the way of our lifestyle, but we need to define what really the sustainability and future of the system. So we talk a lot about the AI, we can generate any number of the things, make our concepts, our ideas, imaginations quite visualizations, right? But is that really true? We try to address the symptoms, but neglecting the root causes. This is what really we want to do. That green growth here is not about just on the computers, but on the land. So this is where I work nearly about the 50 plus countries across the world, but one thing is very common: land degradations, unsustainable practices, and depletion of the natural resources, very common everywhere. Doesn't matter whether America, Europe, or India. This is where we need to redefine the why sustainable is no longer enough. Can you sustain the present world? We have depleted water resources, our food is poison, we not have enough green cover, temperature increasing. Can you sustain there? You see that even sustainable development was 17 UN SDGs we defined few decades ago. Are you there yet? No. Why? Because of you are addressing every SDGs in isolations. We are not interconnectedness. This is what you need to address really. But being a young generation here, we talk about the Atmanirbhar Bharat. India want to become a Vishwaguru. Are you there yet or not? But these figures clearly say that 1 in 2 Indian adults, they are lethargic. Probably we are most energetic, but not the case with everywhere because of the food we eat. 1 in 3, 1/3 of the India, they are diabetic. India became a diabetic capital of the world. It's not just only ending there. We are the more cancers, chronic diseases increasing. You put together more than 70 to 80% of the populations, one or the other way, chronic illness or diseases or disorders. Why is happening that? So this is the broken land systems and broken food systems, collectively called broken One Health systems. You look into all the Panchabhutas, we talk about it. Our land is degraded, our water is become impure, air is polluted, not ending there. Agni, what we get the sunlight on our skin to get the vitamin D3. Look into all the people living in the cities, urban areas, they have the vitamin D3 deficiency. Why is that? Because of a lot of the aerosol particles, they're not reaching the spectrum that reach your sun, your skin is not there. So we lost all the Agni also. I suppose cosmic space, none of you living on the roof. The childhood you lived on the roof, outside, can you see anyone see the sun? Yesterday, full moon, how many you seen the full moon yesterday? Sitting on the roof. I'm glad at least few hands are there. So this is where we lost all the 5 Panchabhutas. So why I put in the health is the wealth? We are addressing the nearly 500 Fortune 500 company CEOs. They're getting the very fat salary, big money, but they're not getting the good food, good water, good life in the cities. That's why we always call health is wealth. We have enough money, but health is not there. What to do with the wealth? So this is where you need to address very critical things in the future. So that's why soil and stomach and gut biome, they interconnected. If you want to do something big, you call it gut feeling, not the brain feeling, because of gut and brain are interconnected. So that's why same with soil biota, health of the soil, health of the human beings, they interconnected. This is what we are facing at the moment. So look into our present agriculture systems, monocropping, completely degraded, depleted, but we need these systems. We need to change completely dynamic at a global level. This is where the world is divided. We see that all the tariff war, future war is going to be the water of the war because of our groundwater depleting massively. Not a single stream left. We can drink water from our hand directly. Everyone need a filtered water. Even the village in the most poverty line, they still cannot drink the river water. That is our real system is there. It is a critical. So this is where we talk about all the 17 SDGs, because topic is today is about science for sustainable future. Are you there yet or not? So this because we are working all disconnected. So this is what you need to bring the more life to the land. That's why more biodiversity in the landscape, in our agriculture leads to more carbon in the soil. More carbon in the soil, it means more water holding in our soil sponge, return of the springs and rivers. That leads to more productivity. More productivity leads to the more equity and social inclusion. That's what. But we are not there yet. The current land use system is completely collapsed. We talk a lot on that. And same is our planetary boundary conditions. So working with the first time Institute of Climate Research, we heard about the Yuan Rockstone. We are out of the 9 boundary conditions, 7 is already exceeded, overshooting that, unsustainable way. And our food we eat and agriculture is not agriculture, not just only food we eat, the cloth we wear. Lot of the even raw material go into the everything you do it that come from the land, but unsustainable. This is where our challenges are there. Look into the industrial growth. We see there from there, we keep on removing the diversity, trees, everything. This is where 2010, there's a baseline I put it. And 2020, that's where I returned back to India. So looking into the where the India is heading towards in the sustainability. Are only economic growth or real growth is happening. And 2030, you wanted 30% green cover in India, especially agricultural lands and land use practices, but you're not there yet. By 2047, we want Atmanirbhar Bharat, self-reliant India. Are you there yet? But 70, you want a net zero. Means anything we do that is a sustainable, economically well, ecologically sustainable, and no impact on the environment. That's what the net zero talk about. Can you reach that? Yes, only possible you can do that. Despite of the business as usual, this is the gray line. This is where we started little bit worry about our earth, our future. Some conservation started here. We still hit the 2-degree temperature. Remember, 4% green cover reduces 1 degree temperature. How much percent reduces to 2 degree? 8%, but still not there yet. But we need to be the at least baseline of the industrial era. We need to back, or you want to make a 1 degree cooler, then you have to increase, bend that curve. It's only possible you talk about the regeneration, resilience, restorations, until you bring the revenue to those people, the farmers, the producers, and the ground level activities. It's not going to happen. So this is the future is where we talk about. India celebrated the G20 presidency. We also put the 2 things apart. G7, G8 countries, largest economical, what you call economic growth countries, progressive countries together, 70, and put the only China, India together, we are the 50, 49% of the economy. Just only 2 countries, 37, and India is about 17% of growth now. Is that really going to address that, the critical mass? This is where nature-friendly farming to the nature-friendly food, it is the 2 things, but health connections and the society. The way we produce, doesn't matter what it is, whether food, whether cloth, whether the computer, are they sustainable? This is going to define our future because of the sustainability in the blood of the Indians. We always try to how to conserve, how to restore, how to respect the nature. That's what India can teach the world. This is the future you can lead the world. So I just give the example of the everyone need a food. Doesn't matter whether software engineer, professor, vice chancellors, and farmer, everyone need a food. I'm putting that as a first conventional systems. Look at that one, all the intensive system. We crossed one of the largest rice produced in the world, 150.8 million tons. You know how much water is consuming that? Can you guess, anyone? It is anywhere between 325 to 525 square kilometers. That equal to 25 Kaveri rivers. And we're exporting 23 million tons of the rice every year. Last year we exported. That equal to about 6 Kaveri, 1 Krishna river going out. It's not just water is exporting, but along with the subsidies going there. This is the massive thing there, but that's why you need to be, take the science, which is biodiversity, climate, food security, value chain, equity, bring the ecosystem sciences, what you call regenerative future. So why is that? In the COVID, you might heard about that. People who are connected to the nature, eating the healthy food, their gut biome is very good, good biome, gut diversity, they never suffered with any viruses. That's where the reinventing their food systems, why the Indian food systems are so good and carbon neutral and low carbon footprint in the world. So in order to bring that, we need to increase the diversity in the food we eating, and that's where you have to build the structure, functions, and production service together. That's the more health per area. This is where you need to focus on that. We often think about a lot of the droughts. Water is not enough, but we did the research very systematically. I'm coming from the Bijapur dry area. Often you always tell that, no, it is very dry area, backward area, no rain, not much enough water. We took that into the scientific research. If you consider my area anywhere less than 500 millimeter rainfall, it reaches about 22 lakh liter per hectare, per acre. But just 1% soil organic increase in the land, it hold about 5 lakhs liters. But in order to grow one crop, we did 20 only, so how much? 2.1 lakh liters. Means water is not an issue. We're receiving the same amount of the rain in the last 100 years, but rain is not holding in the land. That's why issues there. So this is where the tree-based food systems is become a vital forces. And this is the farm, we are going all the food together. So what do we eat in the food in the fasting? When upas madidre yavadu uta madtiri nivella? Hannu hampla geddegenasu alva? That is our food. Often we talk about millets is our food. No. Sorghum is from Africa, bajra from Africa, and navane, what we call a foxtail millet, is from the Italy. But we think that that's our food. No. Only the birds can eat your millets because they have the beaks to remove the husk from the foxtail, navane. But you cannot eat that one until you process it, cook it, then you can eat it. But what you can eat easily? The fruits, the nuts, is the vegetables, is the leaf, is the birds, is the flowers. That is how our anatomy built for that. We always went in the wrong side of the systems. We have to bring it back. This is where real future is, regenerative is, better productions. That means production without impact on the environment, natural resources flow, and important, very nutrition, food vitality. Only Indians understand the chronic value of the food when they grow in the healthy systems, and better environment. We have to restore environment. Without environment, doesn't matter what you eat, it's also not going to work out. Better life. It's not just about making the money, but are you living harmonious with nature? This all together is the future talking about. Antimicrobial resistance, you heard about drug resistance? Because of all the science students and microbiology and the coming from the prestige institutions, drug resistance become very, very important. When a childhood, if you get a dysentery or some antibiotic required, we're taking 0.5, 0.0 micro mgs. Now they come to the almost like a 5 mg, 500 mgs. We are giving equal to the what you call veterinary medicines. Then again, difference. Yakandar drug resistance becomes so important. That has become a very important. Now the West shifting towards healthy diets. They not want to eat the meats. Gen Zs, we always think that they are more extravagant, those things. Look into in the America, 60% drop in the alcohol. They realize that one is not healthy. So this is what the future is all about. And I'm giving the example rice and jackfruit, why I put it together here. Rice story I told you. 1 kg of rice required anywhere between 3,500 to 5,000 liters per kg of the rice. 1 cup of the rice, 300 to 350 calories, empty calories, only starch. And in order to eat that 1 kg starch, 1 cup of the starch, how much drama it goes? You have to put the cultivate land, put the water, herbicide, pesticide, harvesting, threshing, transport, cooking, cleaning. 7 energy losses happens. But the same amount of guava, you can pluck and eat it. You get equal calories, more nutrition, more fiber, and carbon neutral and carbon positive. And no cook, no boil, no oil. That's what the future is about, why our systems are so. The same area where you grow the rice, the half of the room, it's about a 10 by 10, 100 square meter. You grow 1.5 to 2 kilo. The same area you can put 1 jackfruit, produce 150 kilo. So the story goes like this. That's what we need to define what our future is, not about how we go, what we grow, but is that really make a difference for our sustainability. So I compare all those things to educating our minister, bureaucracies. Grain is not just our food, but fruits and vegetables and nuts and wild edibles. Professor Rajesh sitting here, he did a lot of the amazing work in wild edibles in the dry areas. That is amazing one, because without water they grow so much. You might see outside the hattarki. he planted here to show the city people that there is a hattarki kind of food is there. More nutrition your spinach, your greens, your lettuces, but they grow without the water because they harvest water from atmosphere. We cut the soil, remove the hattarki, soil is dry. Go to the North Karnataka cotton belt, black cotton soil, this much of the what's called the cracks in the soil, no water, but they are surviving. Those are our food system, we have to bring it back. So anyway, this is where science, we talk about the AI, automation, machine learning, the statistics, because of our chairman, today's chief guest minister coming from the earlier, Minister of Statistics also, I guess. So he understood what is AI, what is the real. So this is where I started my backyard, in my US backyard garden. You can see that small garden in the backside. We grow 237 fruits and vegetables in that small space. You ever seen the rainbow carrots? My daughter is eating the 7 types of the rainbow carrots. We grow more than so many varieties, but we're not ended there. I put the spectrum to see, understand how that variability there, which is the what you called a atmospheric window and water window. It is not coming. So that's where you can see, understand that signature, that one, which time of the planting, plant growth need the more water, less water, stress is more, can increase the more nutrition, particularly defined, just defining the water. Like that, so many things can be done in the science innovations. So this is where we started like a very early, that almost 16 years ago I introduced the drone. It's not simple drone. Multispectral, hyperspectral drone to understand what is happening in the individual cells in the plant, in the leaf. Can you detect the water stress condition in the leaf? Nutrition level in the leaf, that's what you need to go. In India, drone mostly meant for only flying, what you call in the movie, in the marriages, or spraying some fertilizers. But we need to go behind, this is where the future is there. Think on this deep into the science. So that way started in the backyard, because that's why I'm telling that all the innovations companies, Google, Facebook, anything, they started their garages, because you can observe the things there very closely, what's happening in the plants, in the life. Everyone should cultivate that one, growing one plants in your balcony, in the rooftop, in the home, wherever possible, to observe how the plant behave to every system there. That is where science cultivates. That small things, what I started in the backyard, leads to one of the largest establishing the Earth Observer Integrated System to understand how the behavior of the climate happening in the plant life systems. This is what the Earth Observing Systems, how really helping to understand what's happening in our life system, Earth systems, sustainability. We have a lot to learn, a lot to explore and lead the world in that space. If I talk about this one like a kind of a typical scientist, what is the different components of the earth system and food system, agriculture, horticulture, fisheries, livestock, all the things. It's so complex, but reality, they're so simple, right? Same thing what I put the previous picture. One of the large project is nearly about $270 million funded project. But that translate that into the visualization is simple. Our food is going, our rivers are flowing, everything healthy, soils are healthy, vegetation is very healthy. That's what exactly all talk about. This is what called an entire system, healthy ecosystems. It's not just about the food, it's a culture, it's a provision, sustainability, and can you measure that? As sir just a while ago told that the AI driven system can tell what is happening in your field, what is happening in nutrition level, what is the moisture level, what is stress level, where the markets are there, where you can sell that. That you can do from even like a entire global picture. We have the satellite from the measuring the every day what is happening on the entire earth. Then look into our Indian satellites, IR series, ISROs. You can come back to even individual farm level. The same thing connect with your landscape, agro-ecosystem, farm level crops, even varieties in cell level. My PhD is from mapping the Indian biodiversity using the space. I was before joining, I was a bit puzzled. How can a satellite, I cannot even see the cell in the microscope, but they want to map a biodiversity from the satellite. That is the science. So we did the mapping the India's biodiversity. That's my PhD. That's really fascinated this one, and start looking into all the satellite. So our gurus are here, Professor Yuva Rao and Professor Abdul Kalam. Lucky to have they taught us. And this is where the real science is there. How much innovations we did India, won't believe. What is happening at the individual farm level to the entire global level, we can track. We have so many satellites. And whatever listed is, all the satellites are free data. So what you can look into the eyes in the space and your mobile and the ground data, you can validate what is happening at the system level. I think that's where we need now. So this is where the computer kind of a augmented science. What is happening in the leaf canopy level and water in the system and the leaf level, you can bring them together, you can come out with a common framework that can define what farmer grow, where is the best suited things. I give the example, for example, the sandal is very costly wood. Everyone started growing the sandalwood without understanding whether sandal grow in a particular region or there. That's why every farmer in the loss now. Because of this, because someone growing somewhere and start growing everywhere. We forget the niche mapping. Is that particular species the ecologically sustainable or not? This is where you need to bring the augmentation of all the things. Science, technology, innovation, ground knowledge, traditional knowledge, wisdom all together, put together, then define where the site-specific integrations so that that economically viable, ecologically sustainable. That means any plant you grow there without any intensive care, they grow very well. That's called niche. But we not do that one. If the wheat is important, you grow wheat everywhere. Without, then we started. Our entire agriculture ICU, we put the fertilizer, pesticide, fungicide, all things, but not a sustainable way. This is where the integration is going to happen. Science, technology, innovation, producer, consumer, institutions, all the interlinked each other. In the three framework, we talk about digital augmentations, where particular things can grow well, where the markets are there, where you can export, where the demand is there, and a lot of research and sustainable framework all then bring together. This is what we called a lot happening in the medical science. Earlier, you know that one, right? How our dental specialist, eye specialist, ENT specialist, ankle specialist, you go to America, even more specialist. When the separate thumb was separate. Now they understood that it's not like that. Medical systems is a holistic medicine, meta-medicine. Often like my headache is there, treat headache, but happening with because of our gut. We're not eating the right food, not taking the rest. So sometimes joint pain is there, we treat their joint, but it is happening with our stress level or the food we're eating. So they all a whole body system interconnected. That's why same thing, the production systems, AI happening at the land level, crop, water, air, energy market, global economy. But they put them together, the meta-analysis, there we call inclusive ecosystems. The economy, talking about India wanted to be the largest economy. I think we already reached to the third, fourth, and soon to be third. Or our economy is sustainable. We are living in the healthy systems. Money is not everything. We talk about the trade deals. We just was fortunate to be involved in that. They're very clear that Europe, they want Indian food. It should be sustainably grown. It's a poison-free. It is not at the cost of the natural resources. Then only you buy, otherwise they won't buy. All the Europeans, you heard about our colonizations. They came to India for the spice trade, putting their life to risk to buy Indian spice and foods and medicines. Now we are sending them, they're rejecting because of the poison in the food. Isn't irony? Someone putting their fight the entire life to come to buy the spice. Now we are growing, sending them back, they're rejecting it. This is the real tragedy India facing. We have to correct that. So this is where you are, all the young minds, take into everything that what you can lead this into the science, innovations. Of course, we talk a lot about the AI, machine learning required. Let's make our life easy. When the JCBs or earth movers introduced, all the worker unions, they went strike. The JCB take my job, I not want. But now we cannot do any work without JCBs. It took the time to understand that they freed up unnecessary job. Digging the well when our childhood, it took about 6 months, about so many people. We did now recently, a JCB did for 1 day. That's what we are freeing up work. So much of the workforce we are doing unnecessarily, like 60% of the job in Bangalore is the call centers or service sectors. They're not doing anything on the ground, growing anything, nothing. Just sitting in the chair job. By the way, chair killing more people than any mosquito or anything, because the sitting is also not good. So the same thing happening with the moment AI coming, I'll coming to that point. This is where we have to understand what is happening, the dynamics. You're buying the lot of the land, putting the fencing, not growing anything. A fertile land turning to the investment locked lands, nothing doing there. Like 12% of Indian land sitting there idle. Is that we want to do it? No. So this is where the fingerprinting of the farming systems. Can you guess what is this picture? Anyone? What is this picture? Satellite image, right? So what is basically, what crop it is there? Can you guess? We talk about the rice. It's all the rice fields. Why they're different each other? Because of every farmer is a kind of a fingerprinting. Every farm is unique. We cannot generalize that. Some farmers, they burn their land after the harvesting. Some they are not at all the very dark red, they are not yet harvested, still the green. Some are the brighter, they harvested already. And some are the planting early variety, late variety. Someone is very good managed, someone is not managed, someone is water well, not well, variety is different, weed managed different, all the figures are different, but all are rice fields. But one thing is common, can you guess what is that? All the field bunds, I mapped that one using satellite, drone. Nothing is growing in the field bunds. We removed all the trees from the agriculture fields, thinking that the productivity goes down. But actually it went even much lower because of the harm made per paid, what our prime ministers talk about, but our all bunds are free. And we wanted to do a very interesting project. So I asked him to, every farmer should grow about at least 100 trees around the farm bunds, so that having the trees around the farm as a wind belt, central belt, reduces the output transpirations, leaf fall in the land, increases your soil organic matter, reduces the fertilizer use, and soil, the roots breaks the soil so that infiltration goes, the rivers flow. But they said there is not any land to grow the trees. Then I put into the one small district in Devas, the MP, just took a small district, geographically about 700 hectares, and cultivated with that much. The length of the farm boundary is nearly 300 use that one, use 19% recovery increases what Indian is only 8% increase to reach a 30% goal. So we have to look at a different way how we can see the landscape. So, then, so I have a lot of interesting things to talk about. So we did a lot of the nuclear research and fusion research, what we can do, what we can understand what is happening at the system level, so that monocropping system, make a 6,000, make a 30 or 60,000, like that you understand the cost of the transitions into a deep, this way a digital augmentation, science, technology, questions comes together, along citizen science. As I was talking about, one put a picture on the ground, tell about how much the available organic matter, how available the nitrogen, all the different things you can done. That really helps us to understand where is my land is productive, where the loss is happening, where increase is happening, in the farmability is there, can address that. This is where it really happens. At the same time, running very fast, being the science, so I understand that. So there is the actual landing status in the Iran, Iraq, put them together. And if you change into a little bit of addressing the some changes, we will land productivity, but I can identify where I can put the low hanging areas. Small interventions, if you change that to the area, then we can increase the 5% gain. And this is where you would understand that. These kind of interventions. The roadmap is very clear. Science is there, innovation there, technology is there, citizen science is there. But you have to define the path impactful so that you can lead the world. Lot area degraded and is within long ago, but how you can really receive the needs in technology innovations. And most importantly, they are very fragmented farmers, very small holders, less than 2 acres. But good thing about everyone is so how best we can use them to define. This way I thought addressing the all the together, where they take a great Indians, all the Indian civilization past to the campus, to the country life, to the restoration future is very clear. This is the what I'm telling about that. We went from the agriculture economy, system economy to went to the service sector economy, but yet take most of our job. Young generation, we talk about early career research as you are going to lead. So break the barriers, back to the career monopoly, what I call monoculture career, only engineering, medical things, but basis thinkers and you have a better future. That's what youth are towards citizen science, evidence-based movement that's happening at a very large scale. And this the schools have that really made a change. This is the future, is green. So we were sustainable together and every level, from the traditional transitions, sustainability, and by 2030, we need at least 30% of food coming from the sustainable landscape. But there's a recommendation that our food plate should be more sustainable so that we can lead the back to the future, drive the green technologies, live guilt-free, plan for a better future. And this is where my last statement I'm playing about. Every child born genius. Either parents or teachers made the media. So best universal parents and teachers can do is taking their children to the nature. Nature is the last best. Nature best teacher. Thanks very much. Thank you, sir, for the stopping you. Because of you are the driving the mass crowd. So thank you, sir. Thanks. We are Trimurtis, so Brahma is to Malaysia. Yeah, sure. Sure. One photograph with you, everyone. Okay, yeah, sure. It's better. Thank you. Because of hearing your stories, sir. When? Because of the students that are going to drive the future. You don't think you're better than that. Because we need a lot of students. So let's open the thanks for message. So let's thank you, madam. Yeah. So let's have the keep it very informal. I know a few minutes before we go to the lunch break. Any questions?
Yes, anyone has any questions?
Are you hungry? Okay, that's fine because we have to keep the time. Agree.
Thank you, sir, for such an informative session and showing how integration of AI system like agriculture to the silent view. Now I request Dr. Chandrapal sir kindly present a letter of appreciation to Professor. With this, we come to the conclusion of the second session. We will now break for lunch. Participants are requested to assemble back by 2:30 p.m. Thank you.
Genes are there. So all the genes are most, I cannot say all, most of the genes have been sequenced and its function is known. So it is believed that whatever gene is present in Arabidopsis is also present in the other crop species, maybe with minor mutations. So we can decide. So in Arabidopsis, if a gene is coding for bacterial resistance, we can also confer that gene may also, homologous gene may also confer resistance to bacteria in pomegranate or any other crop. So once alternatively we can go for the differential gene expression analysis. Usually transcriptome analysis using RNA-seq will be done because now the sequencing has become so cheaper. So with just 25,000 we can go for one transcriptome analysis. So we will take the resistant, susceptible or sometimes inoculated and uninoculated, do the transcriptome analysis, identify the genes which are differentially expressed, either upregulated or downregulated. You can select the genes. You can also go for comparative genomics. I think it is similar with the homologous gene expression, or to prove we can also go for RNA interference technology. So in literature already a lot of genes conferring different traits have been published. I think you can go through the UniProt or NCBI or Ensemble plants, you can get that information. So that will solve our lot of problems in identifying the gene. Next is the designing the guide RNA. So guide RNA is complementary to the target sequence what we are going to edit, but it should be adjacent to the PAM sequence, protospacer adjacent motif. So it is difficult to search manually. For that reason several tools are available. The most commonly used is the ChopChop, Synthego CRISPR design, Gene CRISPR, like that lot of tools are there. So what these tools are going to do is first identify the PAM sequence, then identify the sequence complementary to that region, and most important thing we need to do is we need to check, because our target sequence is hardly 20 base pairs. So there is every chance that this 20 base pair is also present elsewhere in the genome. So we need to make sure that no off targets are available for the design bit. So next is the construction of the guide RNA Cas vector. So if you are aware of the vectors, so basically it's an expression vector where the gene has to be driven by a promoter. So we can put the Cas9 protein under any of the promoter. Most commonly used is the Arabidopsis thaliana ubiquitin-like promoter, U6 promoter, or CAMI35 promoter, cauliflower mosaic virus 35 promoter. So we have to make sure that the Cas9 should be driven by a promoter, as well as the guide RNA what we design should also be driven by a promoter. So once it is done, and the advantage of CRISPR-Cas is we can edit multiple genes in a single vector. So this is one example where you can see that there is one gene here, there is another gene, second gene is here. Okay, you can edit multiple genes. Till now, there is a proof that 6 genes can be edited simultaneously in a single vector. So the next challenging part is, so now we have got a DNA vector containing the Cas9 gene as well as the guide RNA. So how to deliver that into the nucleus of the plant? Because the editing should happen in the chromosomes which are present in the nucleus. For that we can go for agrobacterium mediated transformation, where when we clone this vector in between that in the tDNA, it is going to naturally deliver this Cas9 gene as well as the guide RNA into the nucleus of the system. The second method is we can also, if the plant is infected by any virus, so that virus genome can be modified so that we can deliver this one, or else we can go for particle bombardment. In case of particle bombardment, either we can go for transforming both Cas9 as well as the guide RNAs, or if the plant is already expressing the Cas9 gene from other events, we can just transfer the guide RNA. So these all comes under the transgenic events. Once edited, we should make sure that the guide RNA, which is a transgenic, from the foreign DNA, as well as the Cas9 gene has to be removed. So now the alternate approach is, instead of delivering a gene, we can directly deliver the Cas9 protein and the guide RNA. So once editing is done, so these are naturally removed from the cell system. For that we can use some nanoparticle mediated delivery system. Okay, so the next important part, if you are using any agrobacterium mediated transformation or the particle bombardment, we should make sure that a efficient regeneration and transformation protocol should be ready. So this is one example in case of chili. As you can see, so first using leaves, they have regenerated the callus. From the callus, they transformed this callus. So the transformed cells. So one more thing I forgot to tell you, in the vector design, it is advised to put either a selection marker like kanamycin resistant gene or any herbicide resistant genes, and they are going to be selected. Only transgenic events are going to be selected on this selection media, so they can be regenerated and then tested. So once these plants are regenerated, next important step is to check whether the plants are mutated or not. For that reason, we mostly go for the sequencing. So we know the region where it has supposed to be edited. You can design the primers flanking that region, and then you can sequence it. So as you can see here, so when you see that there will be some mutations, as you can see there is a deletion, so there is a deletion, so there is a 16 base pair deletion, like that. So you can see lot of mutations in them. So once the mutations are confirmed, the second part is make sure that apart from the target region, mutations are not created anywhere else in the genome. So after that, you are going to use that event for screening. So now editing has been done. So as you can see here, there are a lot of edited lines are there. But which one has the favorable addition or the favorable mutant? So that has to be confirmed only by the phenotypic screen. So next important thing is, if you have used any vector mediated delivery of the transgenes, most important is to remove that guide RNA as well as the Cas9 gene. So that can be done by, so in the T0 plants, which contains both the guide RNA as well as the Cas9 protein gene, and also the edited lines will be there. So they can be selfed. So in the following generation, T1 or the T2 generations, you select for an event or the plant which is edited but does not contain any transgene of guide RNA or the Cas9 gene. So those can be selected by. Alternatively, as I told, instead of going for the DNA vector, we can directly add the CRISPR RNA as well as the Cas9 protein, which is called as the ribonucleoproteins. So as you can see, there is a Cas9 protein and then the guide RNA, both of them are clubbed together to make a ribonucleoprotein, and with the help of several delivery methods, most commonly used is the nanoparticle mediated delivery. Alternatively, if you are able to isolate the protoplast, so directly these can be delivered into the cells and regenerated from them. So this is one example why RNPs are more effective. So it's because it is just, I call it as hit and run case. So you go, edit and disappear. So there is no transgene left in the host system. And it has got a lot of reduced off target. By chance, if the Cas9 protein, the gene coding for the Cas9 and guide RNA is there, there can be lot of events happening over generations. Sometimes that can lead to off targets. But in this case, since it is just hit and run case, there is no chance of off targets. And many times in the vector mediated or the DNA delivery system, the Cas9 gene can be inserted anywhere in the genome. Many times there is a chance that it can insert into some of the functional genes that will lead to the insertional mutagenesis. By using RNPs, ribonucleoproteins, there is no chance of any insertional mutagenesis. And so if you can if you are able to regenerate from, isolate the protoplast and regenerate from the protoplast, there is no chance of cell wall or the membrane barriers. Directly they can be delivered there. Okay, so this is the whole story of editing. So once the editing is done, although we say that the gene edited plants are deregulated, but they have to go through the process of regulatory approvals. So usually it starts from, so as I told already, there are 3 events, SDN1, SDN2 and SDN3. So SDN3, which still retains a larger sequence, it has to undergo the same trials or the regulatory approvals as the transgenic crops. Whereas rest of them, no need of them, but it should always start with the IBSC, Institutional Biosafety Committee, which is there in the institute. It has to be approved by them. After approval, it has to go to RCGM. Then in the RCGM, so there will be several layers of regulatory agencies. They keep verifying your data. The data mainly includes, check if there are any foreign gene in that events what you are going to deliver, and if there are any unintended mutations, so that all you have to report. They might ask us to submit the whole genome resequencing of edited line in comparison with the original line. Sometimes they might ask for metabolomics and the proteomics data to check if there is any unintended mutations happening with the metabolites and the proteins. Then only it is going to be approved. So now the average time for regulatory approval of gene edited crops of SDN1 and SDN2 are just 2 years. So that means within 3 to 4 years, our gene edited plants are going to be reduced. So with this, I'd like to say that, so this gene editing is a bigger tool for us to achieve the first 3 sustainable developmental goals, that is no poverty, zero hunger and a healthier nutritious food. Okay, so as I told earlier, so already it is not a new thing. Unlike transgenics, lot of restrictions are there for the release of transgenic varieties where they have the foreign gene. from different countries, but in most of the cases, if it is green, it is already approved. If it is yellow color, where they are making the policy changes, they do not allow the direct events, but they allow the import of gene edited crops. So only the in the red one, only SGN1 are allowed, rest of them are not allowed. So it says that lot of crops have been already approved, gene edited crops have been approved to achieve our sustainable developmental goals of 1, 2 and 3. Okay, so this again there are some sometimes if the gene, the Cas9 gene is in the system, it can also make some off targets. To overcome that, there are some recent developments. The first one is called as the base editor. In this case, so there will not be any insertion or the deletion, but just a target nucleotide is edited. So by using two proteins, extra proteins called as cytosine base editor and the adenosine base editor. This is another enzyme part system clubbed along with the Cas9 protein, that is called as the Cas9 nickase. So what it does is, so the cytosine base editor are going to change from adenosine to inosine, and then further it is changed. Similarly, cytidine is changed to uridine using this. So these are very precise. So in the target region, they are going to change from cytosine to the adenosine, and then adenosine to the adenine to the inosine. So they are going to make only those changes, and they are very precise. The next one is the prime editors. So in this prime editor, so since SGN3 is not allowed to be released, but we can introduce an RNA. So editing happens at the RNA level with the help of the reverse transcriptase enzyme, which is delivered along with the Cas9 system. So these two systems are highly precise, and they are being used, but till date, since they are in commercial stages since last 2 years, so no varieties have been released, but they are one of the good targets, or the good method system for editing the precise, making the precise editing. Okay, so with this, my lab, my team works on editing the susceptibility genes in pomegranate for bacterial blight resistance. So bacterial blight is one of the major disease in case of pomegranate. It causes up to 70 to 80% losses. Sometimes it is 100% losses. You might have seen that splitting of the fruits or the scabs on the fruits, that is the major problem. So all the varieties, currently varieties, if you are eating any pomegranate, naturally you can think that it is the variety called as Bagwa. So this is the 90%, it is distributed throughout India, but it is highly susceptible to bacterial blight. So to edit this one, so what we did was, first we wanted to identify the target genes. For identification of target genes, first we wanted to know which is the pathogenicity factor. For that reason, we went for long read whole genome sequencing of the bacteria Xanthomonas citrae Pathora punica, which causes this disease. Then we predicted the tail proteins, that is the transcription activator like effector proteins, because those are the major pathogenicity factors. The beauty of these tail proteins is they act like a plant transcription factors. They enter inside the nucleus of the plant, they bind to certain genes and activate the plant genes. And what are such genes? Those are the susceptibility genes. So one such examples are called as sweet genes. Sugars will eventually be exported transporter proteins. So what is the general role of sweet proteins? So they pump the sucrose from the cytoplasm to the wherever it wants. Among them, there will be one or two proteins which pump the sucrose from cytoplasm to the apoplast, the space between the two cells, where the bacteria feeds on it. So now what this tail does, go and bind to the sweet gene, act like a transcription factor, make it to express and pump the sucrose from cytoplasm to the apoplast, and it happily feeds on that one. So now we want to interrupt that part. So this is the genome sequence. We identified the tail proteins by sequencing. And then these are the tail proteins. Basically, there are 7 tail proteins in the genome, and then among them we identified that tail 2 is the most problematic one. So now further we identified its targets. So using several bioinformatics tools, means what are the genes it can go and bind to. Then we used so we used the transcriptome analysis among the proteins which are targeted by the tail, which are differentially expressed when the bacteria is infected. So with that we identified 865 gene, and finally we shortlisted 5 genes as the susceptibility genes. Among those 5 genes, 2 of them are the sweet genes. So now we are editing the promoters of these sweet genes in such a way that the Xanthomonas tail doesn't bind to them. So this is our work. So this is the sweet genes I was talking about. So basically there are, I think I already told about the story, like how they are involved and all. So we need to stop the tail to bind to the promoter of that sequence. Okay. So this is what I wanted to present for you. I'll be happy to answer few of your questions. Of course, many of you made me a great teacher. It's not the fault of, I don't know, it may be a fault of me, or maybe the food provided by your team. Okay, anyhow, good morning. Yeah, if you have any questions, I'll be happy to answer. Sir.
Yield, isn't it?
Yes, sir.
So yield is a quantitative in nature.
Exactly.
It's not
sir.
by single
Yes,
gene.
Sir. Yes, sir.
How we could able to boost that one?
So that is really interesting story. Maybe lot of people ask the same question to the one who developed this one. So that gene is basically a gibberellin related gene.
Yeah, but
so it modulates the hormonal levels, and which indirectly regulate multiple genes which are required for the yield. So that paper is in the pipeline, sir. They are planning to, they have submitted to some Nature Biotechnology also. So they could not reveal the much. The one thing they told was, it's a gibberellin related gene which activates multiple transcription factors.
That means pleiotropic in nature?
Yes, sir, it is a pleiotropic in nature. So it is going to activate multiple transcription factors, and transcription factors in turn, I think as per Satyendra who developed this one, there are around 67 genes which are differentially expressed when this editing was done. So it is, as you are true, it is a QTL, sir.
Amazing. So in your project work,
yes, sir,
you are targeted for tail proteins.
Yes, sir.
Which are having transcription effect.
Yes, sir.
And they will make sugars pumped into
apoplast.
Apoplast.
Yes, sir.
But you know that transportation of sugars.
Yes, sir.
Through 3 mechanisms: apoplast, plasma desmata, and
yes, sir.
some others. Through three mechanisms, molecules move.
Yes, sir.
If you block this one,
sir,
because of
sir, through all the three mechanisms, the protein involved is sweet. Whether it passes through plasma desmata or directly through the cell pores, but it is through the proteins. Those are the sugar carrier proteins.
Yeah, but then whatever the plant itself, because if you block it.
Yes, sir. This is a nice question, sir. I think we have been addressing for last 2 years. So we looked into the genome of pomegranate, and we found that there are 24 sweet genes are involved. So among those 24 sweet genes, we tested their expression at different conditions, just without any infection in the leaves, in the fruits, and after pathogen infection. We found that there are different sweet proteins are involved in each process. The proteins which are responsible for sweetness of aerials are different from the which are involved in the photosynthesis. And we identified 2 sweet proteins which are involved, which are upregulated only when Xanthomonas is infected. And we designed the guide RNA in the 3' UTR so that other genes are not suppressed.
Okay, so many are there. Probably the sink, where they are going to pump the sink, that's a very vital for our
Yes, sir.
food.
Yes, sir.
Probably they are different from these.
Yes, sir, exactly.
Yeah.
Yes, sir.
So that is
Yeah, we are taken care of that, sir. Thank you.
My question is like related to in case if editing the target is the sweet genes, but it can be eventually the changes in the xanthomonas also in the future.
That's a good question, sir. I think so. Sir, question is what happens if the tail changes its binding region? It is expected. So that's why we are not doing editing only one part. So we are editing the multiple parts in the promoter region. That's one thing. Apart from that, we are also targeting other defense related genes like receptor like kinase. We found that receptor like kinase, one receptor like kinase is highly upregulated when the pathogen is infected, and it activates several other downstream defense mechanisms. So we are targeting multiple genes. That's why I said 5 genes, so that the xanthomonas will not overcome these mutations. It is expected, sir. So there is a nice story I didn't present. It's called as executor R genes. There is another method. So now we can confuse the tail. So design a tail which can be bound by the tail to the R genes, that is the resistant genes like receptor like kinases. So now what it does, it goes and binds. Instead of activating the susceptibility genes, it is going to activate the defense related genes. So that one part also we are taking up. So multiple approaches are taken.
Thank you, sir. And
Thank you.
another question is like this editing is in the developmental stages of the cells. CRISPR, and if it is already differentiated cells, is their approach feasible?
Sir, usually like DNA replication has to happen. So then only it can be multiplied, so that multiples. So usually we edit in the single cell. So before the, before differentiation, so that the single cell will divide, then attain the stage of tissue, then tissue is developed into different organs, so that from the single cell whole plant will be regenerated.
Sir, for example, like tissue-specific stem cells, which may transform into malignant cancer stem cells and carcinogens in that cases.
Yeah, fortunately in case of plants, all the cells are malignant. So we can, totipotency, every cell is a totipotent in case of plant cells, whereas in the human beings it's only stem cells. So we can take any part and regenerate it into a complete plant. Of course, we need to play with the different hormonal levels to make it right protocol.
Thank you.
Yes, sir. So another one mechanism we are working is, so now the pomegranate archers are already developed. So can I edit them? So I think one of my students who is here, she is going to work on that one using nanoparticles. She is going to deliver the RNP particles as a spray method, like RNAi sprays. Yes, sir. So if it works, then we can use them for the several year old orchards also. So that doesn't require any tissue culture. Only for that generation. Yes, sir. Only for that plant. Yeah, that can also be done. Transgrafting can be done. Yeah, it can be done.
Thank you, sir, for such an insightful and informative session. I now
Thank you.
request Dr. Vasant K. Baskara, Department of Biochemistry, to give the appreciation letter to Raghavendra, sir.
Thank you, sir. Thank you. My pleasure. I thank our Director of Research, Dr. Fakhruddin, who was supposed to come for this lecture. So he could not make it because of his busy schedule. So I thank the organizers for giving me this opportunity. Sorry, I didn't present about one thing. So we do provide internship opportunities or 1 week training programs in 5 different modules. The first one is the gene cloning, including the gene editing. The second one is on DNA fingerprinting of plants with several molecular markers like RAPD, SSRs or SNPs. The third one is on plant tissue culture. And fourth and fifth are in bioinformatics. Fourth is the basic bioinformatics like sequencers, tools, and phylogenetic tree construction and all those things. The fifth one is on RNA-seq analysis. So if any students are interested, you can contact me. Thank you one and all.
There is an announcement for the participants. So tomorrow there will be a quiz based on the lecture series, and it will be 2 in a team. Those who are interested, you can give your names to the registration desk, and you can get it registered. Now we can have there is high tea that is arranged. I request participants to join us for the high tea, and register your names at the registration desk now only. Hello. Uh, so tomorrow 9:30 you have to assemble in the same auditorium for the second day of the lecture series. Thank you.
Students, here is a small announcement. Certificates will be given to candidates only who attend all the sessions. Okay, kindly note. Certificates will not be given who will not attend all the sessions, and the certificates will be issued tomorrow in the valedictory session. After that, you cannot come and ask us the certificates. Okay, so you have to be present in the valedictory session, only then you can collect your certificates. And students who want to participate in the quiz, ma'am has already given you the insights of the quiz. You have to register today itself. No registrations are taken tomorrow. So the registration desk they are sitting, you can enroll your names for the quiz. And only interested students can enroll, and no registration tomorrow. And the quiz competition is based on the first 5 lectures. From today till tomorrow, the first 5 lectures, it will be covering 40 questions, and it is a MCQ written test, the quiz. And there will be 2 members per team. Okay, 2 members is a must for the quiz. So you people can pair up, and 2 of you can give your names in the registration desk. And it is for half an hour, and the winners will be assessed based on the questions you have answered for all the sessions. Okay, so from each sessions around, I think 10 questions or something will be given. So it is, the assessment is based on all the sessions how you answer. It is not like only the 4th session you answer all the 10 questions and the rest sessions you don't answer, you will not be considered. Okay, your answers will be considered based on all the 5 lectures, how you answer. So this is how we are going to evaluate your quiz. And the winners will be given cash prizes along with the certificates. Clear? And tomorrow you have to assemble sharp by 9:30, the same venue.